Yes, so welcome everybody for the third day of our annual national meet, where we have hundreds of students across the country who were working collaboratively on numerous projects, presenting their work and debating and discussing and actually playing the game of STEM, which is practicing science and making things. Before we begin with the activities for day 3, we just saw the video of chatShaala.in. So it's a small platform that we have developed at the Knowledge Lab at HBCSC, where students can have threaded conversations across the country and collaborate on various projects. Please log in to the in the links that are provided, all the participants in the YouTube as well. In the descriptions, you can see chatShaala.in. So you can register there and log in and find about hundreds of projects that students are already working on. And without making any further delay, we will move to the activities of today's annual meet. And there are lots of sessions and lot of surprises. It's going to be a fully power-packed day today again. And so to begin with, we will have a quick reconnection to what we were talking about when we ended day 2 yesterday, last night. So we were discussing the microscopy. So many students here have been using a lot of ways of taking beautiful pictures. For example, if you can see, one of them is right here in front of our eyes. So this is a beautiful picture that the students have taken of a tiny fly, which is a fruit fly, its wings. And so we are going to continue for about 5 to 10 minutes about what we are planning to do with the microscopy, and then we will start the sessions, and there are going to be some surprises also in between. Please stay tuned. Over to Ashish, Ravi, and Jude. Yes, Ashish.
Yeah, hello everyone. I hope
Hi.
I'm audible. Yeah.
Yes.
Audible, yeah. Yeah. So I mean, it will be really honor for the makerspace to actually announce the collaborative. I mean, we invite the collaboration from the Cube and other participants also to work on the microscopy.
Yeah.
So the idea is how we can actually do the tinkering to come up with some frugal DIY microscope, which will actually help us in doing experiments or watching microorganisms in our home labs.
Yeah. So yesterday, Ashish, we had a brief conversation. So the students are already using their mobile phones, and they were able to capture even sub-millimeter, that is almost 0.4 mm size of things using simple mobile phone, and some play with the light and shadows. Are we going to briefly touch upon those aspects also to begin with, to understand what all are the capacities of our mobile phone?
Yes, GP, definitely. We are going to explore everything through which we can come up with a microscope. So we will start with the mobile phone. We will see what are the different approaches that we can make our smartphone as a good microscope for our home lab use. Yeah. So we'll be looking how we can play around with that light around the capturing the microscopic images, and also what we can do to actually take those images, adding the macro lenses or adding other things to our mobile camera or smartphone camera.
Yeah. So actually, yesterday at the night we gave a simple task to some of our participants to sort of figure out the limits or the capacities of the cameras on their smartphone. We asked them to find the focal length of it, what is the smallest area that their phones can capture, and we asked them to also post some pictures. I think because of the tight schedules or whatever, like we haven't been able to gather any responses yet. But I think after this event, that is after today's activities, I think definitely we will have more interactions moving ahead with the students. And do you want to talk about the share my screen if you want to? And Jude and Ravi, please feel free to join in the conversations. So we will, in the next 5 minutes, we'll close this here.
Yeah.
Okay, so you want me to share my screen?
Sure, DP.
Okay, sure. Yeah, so is it visible? Yes.
Yes.
Great. So as you can see, we are planning to have a series of regular interactions where we speak. Okay, so someone has taken away presenter from me. Sorry for that. Yes, so we are planning to have a series of sessions, regular interactions, where we discuss things about microscope and microscopy. That's why it's called DIY microscopy, do it yourself. So this is part of a series of other activities that we have been doing, say with charger or speedometer kind of things, which are basically about tinkering with various things which are very easily available around us. But I think this is a very first thing where we seek collaboration with lots of biology students here, because they are already doing a lot of microscopy. And yes, does anyone else want to add something here? Yes, Ashish. So when are we going to have, and can you give more details?
Yeah.
Yes.
Yeah, sorry for the pause. Actually, I was speaking and yeah. Okay. Yeah, so we'll be starting this session. From 12th November every Thursday during 3:30 p.m. to 5:30 p.m. And we have not yet decided how many number of sessions will be there, because we want to go slowly and cover all the fundamental around the microscopy. And so that when we will actually talk about the development, and when we will get our hands dirty while the development, so all these fundamental will actually help us to come up with some good a good frugal DIY microscope in our hand, so that can be used in the home labs.
Yeah. So I think we are working very closely towards setting up home labs, more and more capacities to the home labs using things which are around at our homes. So that that's a part of this series of DIY activities. Jude, do you want to add anything to it? Is Jude here?
Yeah, I think he's here.
Yes.
Yeah. Audible?
Yes, you're audible, Jude.
No, nothing more to add.
Okay. So Jude is also one of the mentors who has been supporting us in these activities, and with a lot of experience in industry. And I think we are going to have a lot of interesting conversations. So we invite all the students in schools as well as colleges who are currently among the participants here. So please, we will share these links to all of you, and let's have an additional conversation or chat-shala around the microscopy. So it's going to be dedicated to begin with, where we speak about microscopy, and we will see, as Ashish mentioned, we don't have end to it. We will see how the conversation builds, and we will try to connect it to all your contexts.
Sir, so is this DIY microscopy similar to foldscope?
So this is foldscope is also part of DIY kind of a thing. We can discuss foldscope as well here, but this is going to be much more than foldscope, where something we are going to build microscope. We are going to understand what's there in the behind microscopes, and in the process we understand how well can we make use of the things around us at our homes to the best of their possibilities. Yes, foldscope is the magic. Yes,
definitely, yeah. So just I want to add one point, like it will be more focused towards like building your own digital microscope. So as we have the power of computers in our hand, that is our mobile phone, right? So
yes.
like all the processing related to the image can be done by that device.
Yes.
So now what all we want is just to capture those microscopic images. So the you simply capture it, you take it, you can also process it, and you can share it in the same moment.
Yes.
So so we will be like focus on like how we can capture the microscopic images, how we can increase the resolution, and how many what kind of how many times of magnification we can achieve. Yeah, we will cover all of these fundamentals during the sessions.
Yes. So we see some participants on YouTube also who are excited about it. So we will post the links for these sessions in the description as well. Please feel free to share this among your friends and your colleagues who will be interested in this. And it's going to be like our regular chat-shalas, where we have lots and lots of debates, where we get into details of what's happening inside. So we leave no box closed. So we will open everything and sort of figure out what's happening inside it. So that's the spirit with which we are going to have these sessions. So I just pasted the link on YouTube Live as well. So you can get more details on that particular link.
So this will aid our home lab also.
Yes.
Many students are working on such minute projects.
Yes.
So this home lab DIY digital microscope will actually help us a lot. Thank you for this.
Yes.
And also, what are the things we needed for this, like for making this DIY microscope? Is it something we need to buy as a to attend the session, or is it all available things in our home?
Yeah, so we will start the conversation this 12th of November, the coming Thursday, and we will get a sense of what all are available around us and see on how we can make best use of them to get the best microscopy possible at homes. So so it's basically it would involve a lot of tinkering around and figuring out the material capabilities of various stuff that we see around us. Yes. Yes, mostly with smartphone and from things around.
Yeah.
And we will slowly build towards developing some sophisticated microscopes as well. We'll move towards that. Yeah.
So, in case of webcam, you might require 2 mm or 1.5 mm fine screws of length about 25 mm. So that's a bit of a specialized requirement, but it should be available in most cities or most tier 2 towns as in the hardware stores. So that is one requirement that is going to be there.
Yeah. And perhaps we could find them in some of our old gadgets that are around. Can we, Jude? Those screws?
Unlikely.
Okay,
because,
okay.
Screws of this length are not usually in most of our gadgets.
Jude, your voice is low. Could you be closer to the mic? We are not able to hear you properly.
Is it clear now?
Yes, better.
Yes, better.
Better. And is it clear now?
Yeah.
Okay, I have two microphones, so I'm unsure which one is.
Yeah, this is perfect. I think we'll have to quickly conclude so that the other sessions also can start. So please say what you wanted to say very briefly, Jude. Yeah.
Yeah, so the statement was that a specialized screw in case of using a webcam as a microscope is required.
Okay, okay. Yes, so we will figure out various other details when we meet this Thursday, and we are looking forward to seeing you all this Thursday. Yeah, that's it for this, and let's move with the other activities. Over to Meena.
Okay.
Yes, Meena, you are very much clear.
Fine. Okay, thanks, DP. So welcome to the day 3 sessions. So we are going to first begin with the presentation of earthworm, and then it will be followed by presentation on hydra. So I mean we are adjusting the time, so it is nearly about 5 hours and 50 minutes. So you'll have 40 minutes. So we can have this presentation say about 6 till up to 6:30 p.m. Okay, so over to you, Earthworm Group.
Yeah, so I'll upload the video.
Yeah, and we are taking questions in this webinar, that is BBB chat, and we encourage the YouTube audience also to please post their comments, questions on the chats, which will be addressed here after the video. Okay. Okay, so nice video. So other group members here joining, they can unmute themselves.
Yes, ma'am.
Ritik is here, Sachin, Lokesh. Lokesh, any questions, comments from the YouTube viewers? Please post
No,
them or
ma'am, but
tell us.
the video
Yes?
was amazing. But there are some questions regarding your setup, Ritik. That
Oh.
why Ashutosh sir is asking that why don't you culture with the soil?
Ah,
Yeah,
okay. Okay.
why
Hello.
not directly
Hello.
take the soils in the cup?
Hello.
Hello.
Hello. Am I audible? Hello.
Yes, yes, you are.
Yeah. So why we are using tissue paper is because observation is quite easy, and when we are looking at tissue paper which has white surface and hello, hello, hello,
Yes, yes, you
hello.
are audible.
Yes, Sachin,
Am
you
I
are
audible?
audible.
Yeah. Okay. So basically culturing why we are culturing it in tissue paper is because tissue has white surface, and earthworms can be seen easily in that tissue culture. And why we are not using soil is because soil is tedious to work with, and soil can be used for other can also be used, but our main focus of culturing was our main focus was different. That's why we used tissue culture, which was easier and affordable for everyone.
So why not directly put them in the plastic cup?
Hello.
Hello, without the tissue paper, that is, it would be more visible. No? It's a transparent cup that you have.
Yeah, it will be more visible, but what will they feed on? So tissue paper has high amount of.
So are you adding the tissue paper for the major reason is for the visibility or for the feed?
No, no.
I'm a bit confused now.
Hello, hello.
Yes.
Okay,
Earthworm would be feeding
Sachin,
on
I'll take
tissue.
over. Sachin, I'll take over. So we can use tissue paper and we can use soil. So there are two methods of culturing earthworms, but our research question requires a different kind of culturing. So for example, our research question is like my juniors are doing gustatory response and I am doing ventral nerve cord regeneration. Sometimes I introduce a cut in the earthworm. So sometimes it is easier to see if there is any bleeding in the earthworm, and that would be very easily seen in the tissue paper. And the tissue paper is playing two roles. Okay, so first of all, it is helping the earthworm to eat the tissue paper because it is a pure source of cellulose. As we all know, that tissue paper is made up of trees. And see what happens is ki when a person is doing vermiculturing, then that person can think about culturing them in the soil. Okay, but our research question and our focus was different. So that's why we took the cup culture, tissue cup culture method.
Okay.
So one more question is there. So why are you using only muslin clothes to cover that cups?
Or it's just a random choice?
We can use any cloth. We have used muslin cloth because it was available in our lab. We can use any cloth to cover. The thing is, the cup cultures must be covered so that the earthworm couldn't come outside anytime.
So is this a tissue cup culture or tissue culture of earthworms?
If we are using a cup, it would be called a tissue cup culture. We can use any other thing. It is not compulsory that we can use a tissue. You can use a
You
cup.
can even call this culture as tissue bottle culture. So what
Yeah.
happens is ki, plastic cups are now banned. So now people can use the plastic bottles that are there at their place. They can cut them in half and use that instead of using cups, plastic cups.
Wait, can you elaborate it, because the YouTube viewers are asking that the earthworm eats the tissue papers or they get the nutrition from that?
Okay. Um, So what happens is, like we can see in this slide here, where we are culturing earthworms. So it is, one thing is that the cups are very cost expensive. The tissue paper is very inexpensive, and you can find these things at your home very easily. And instead of a muslin cloth, if you use a normal cloth, that would also be fine. So these setup is very easy to make at home.
How about using newspapers?
In them. So we don't know if ink affects the earthworms or not.
But when you say tissue culture, I get a bit confused because the animal tissue culture is also called tissue culture.
We can call this as tissue paper culture also. So it will be that we are using this paper.
Or we can normally call it just cup culture.
Deep is asking that does the ink affects the earthworm that we use the newspaper?
Actually, the ink must be having some chemicals in it, and earthworm may easily get affected by the chemicals. We know, so it will be better to use tissue instead of the newspaper, because the ink may get affected. Earthworm may get affected by the ink and may come out of the will try to come out of the cultures. And tissue paper doesn't have any chemical, some chemicals and something which will harm earthworms. So we are using a tissue paper instead of newspaper or any other papers.
I have a question that we often call that earthworms are the friends of farmers, and farmers use farmers in their farm use fertilizers. So when those are not those fertilizers, can't the earthworms?
So I'll add on to this, Rishan, that what happens is, yes, earthworms do run away from chemicals as it irritates them. So yes, many of our behavioral assays show that earthworms are running away from the chemicals, but they can tolerate some amount of chemicals, but they don't tolerate a huge concentration of them. So actually, we had a talk with Nisi ma'am that we can use earthworm as a bioindicator. So if in a farm there are too much of biofertilizers used, so that will show us that earthworms, if earthworms are not there, like we said that the earthworms avoid the huge concentration of chemicals. If the earthworms are not there, then we can say that the soil is polluted with chemicals. So the earthworm can also be used as a bioindicator.
So, Vaibhavi, when we talk about chemical, everything around us is chemical, right? As in soil, like what Drishant was adding, in the fertilizers would be chemical. So, how are you using that as a bioindicator? I didn't get that point. Can you elaborate on it?
Okay. So, Nisi ma'am, you want to?
Yes, thank you. I will talk. Sorry. Hello, good evening. See, bioindicators actually, they are substances. I hope I'm audible to everyone. So bioindicators, they are present or they are absent. Either by their presence or absent, they are indicating the concentration of certain kind of pollutants or certain environmental parameters or certain things. As in the case of our model organism earthworm, they will be absent. They don't, since they don't like light, and they don't like the concentrations of pesticides and herbicides. Mostly in those areas, they'll be absent in fact. So, but since there are so many different species of earthworm, we cannot generalize this earthworm as a whole. So, depends upon the species, they will be present. So here in this case, they are absent where soil is polluted.
Okay, there are some things. So somebody on YouTube is asking, what is the research question and how do you maintain earthworms at home lab? We want to stress this home lab kind of thing because we all have been working from homes. So this was an interesting question. So maybe the students can elaborate on this.
See, I would like to say one thing. There are two methods. One, as Vaibhavi and the others have explained, you can culture these earthworms in cup culture by keeping this tissue culture. Another one method where we are all working with the vermiculturists, we can use this vegetable waste as well as tea waste for culturing this earthworm. So there was a question, I suppose. So that is also one method. In our home, we can use this tea waste for culturing our earthworm in a separate pack. In fact, we can use a plastic box. We can add this thing, adding sufficient moisture. We have found out that they are actually increasing their number. That is also possible. And in fact, these earthworms we were using for vermiculture.
Can anyone elaborate on the research question? Why are we keeping this earthworm?
Yes.
It's a constant question we're getting on YouTube.
Yeah, a related question about the research question. Yeah.
So, hello, am I audible?
Yes, yes.
Yeah, we presently are working on two research questions. One is to understand the decision-making factor through their gustatory response of the earthworm, and the second is to study the nerve cord regeneration.
Do you want to show that slide?
Yeah.
Okay.
For talking about the decision-making factor in earthworms through the gustatory response, we are performing a gustatory assay. We have shown in the video that how we perform. We take two filter papers and are kept on the plate, and you can see in the picture that one side of the filter paper is wetted by NaCl, and the other paper is wetted by distilled water, and the earthworm is introduced in the space between, and the earthworm is left to see to which side it prefers. In the video, we can see that it prefers the water side by testing the NaCl side, and it moves towards the 100 water side. And we have performed this gustatory assay in 5 sets. Can you please show the sets of gustatory assay, the slide? Yeah, so here we have performed this in six sets. The first we use 100 millimolar versus water, and what we have observed that the earthworms we are performing 30 runs of the earthworms, and we saw that out of 30 runs, 10 earthworms went towards NaCl and 20 earthworms went towards water. In the next set, 200 millimolar versus water, we can see 5 went towards the NaCl side, means 200 millimolar, and the rest 25 went towards water. And in 300, only 2 earthworms went towards towards the NaCl, and the rest went towards the water. So this we can conclude that the earthworm mostly prefers the water side. It cannot tolerate the concentration about 300 millimolar. Till 200 millimolar, it is little tolerable for the earthworms to prefer to live inside. And about 300.
Why did the 10 earthworms go to the NaCl side in the 100 millimolar versus water?
Because 100 millimolar, they is not that saline concentration that will affect earthworm. Maybe that is why it went towards 100 millimolar, because we see at 300 only 2 earthworms went, maybe because of some species difference, because as we were using native earthworms, so there must be any tolerating capacity of other species. So only 2 of this went towards 300, and we can
this NaCl kill the earthworms. Earthworms.
Hello, you are not clear. Sorry, can you repeat the question?
Yes, actually, what is actually this NaCl do on these earthworms? Does it kill them?
Well, NaCl. So earthworms, what we see, earthworms breathe through their skin, and they have a layer of mucus on their body. So what salt does is it performs the osmosis. If salt is in higher concentration, it performs osmosis, and in higher concentration, the earthworm may also die. So this is how NaCl affect the earthworm.
Have you performed this also to say that NaCl actually kills the earthworm?
Yeah, of course. If you kept, have you ever tried putting salt on earthworms? They die. We see. So we can say that the salt kills earthworm.
How?
So it is more like not killing, but it's more like it's preferring the water side more than the NaCl side. Don't you think? It's not dying because see, I can see that 10 out of 30 earthworms are actually going in your 100 millimolar solution.
Yes.
So what do you
Or
conclude
can
from
we,
that?
can we say that because if we add a lot amount of salt, the liquid components will come out from the skin
Yeah.
surface or something like that?
Yeah.
Can we
Yeah.
talk about that as well? Because that can back up your what you are claiming.
Yeah, actually, this is what happens in earthworms. The high concentration of salt, maybe about 300 millimolar, we can say, the liquid components of the earthworm may come out of their body due to osmosis, and they will die. Obviously, they will not survive if all
Yeah,
water
then you
comes out
can
of their
say,
body.
yes, then you can say that what I think Arunima was asking, like the earthworm will die if we put excess amount of soil,
Yeah.
excess amount of salt.
So we have a question from YouTube saying, why are we using salt and why are we using NaCl?
Because NaCl in our lab, you can use any other salt or chemical. It's your choice. As NaCl is easily available in our lab, so we have used the NaCl.
So why the worms run away from the soil during the rainy season or during the rain? Is there a percentage of salt in rainy water?
Yes. So yeah, you want to answer this?
You
What I have read is that the concentration of salts in rainwater is around 150 to 200 millimolar. That is the concentration of salt in the rain. Okay, and that's why when that rainwater gets mixed with the soil, what happens is the soil salt that is there in the soil also gets mixed with the rainwater, so making the water more saline. So you can say that the saltiness of the ponds, small ponds that are made in the soil, might be of 300 millimolar if we add both the concentrations. So that will end up the earthworms to leave that soil and run away from the soil.
Webavi, what do you mean by millimolar?
Hello?
Wasn't audible?
Yeah.
Okay.
Yeah.
Ma'am, millimolar is a concentration gradient. So for example, I am writing on the board, okay? So if we have 1 liter of a 1 molar solution in which, so we have taken the molecular weight. So what is the molecular weight of NaCl is 58.4 grams. No.
So, to make a millimolar solution, ma'am, we first make 1 molar solution. So, for that, the molecular weight of NaCl is 5.84. So, what we do is in 1 liter water, that is 1000 ml, we take 58.4 grams of NaCl. That makes 1 molar solution. And then we make, then if we want to make one.
Yeah, you need to explain why 58.4, why not 23.5?
Okay.
Is that some magic number? No, na?
No, ma'am.
Yeah,
So
there's a reason. Anybody from the group can help. It's to do with the some kind of weight of the NaCl, right?
yes, ma'am. So for sodium, so we will take molecular weight of sodium and chlorine, and then we what we do is we add it, and then that is how we get 58.4 grams.
Right. So you have written 5.84 gram.
Okay, that is
okay.
Maybe can you make it 58.4?
No, you write it. You scratch that and write afresh.
Oh yeah, correct.
So
That means that is
Yes, ma'am. Ma'am, you are muted.
that means a very low concentration of salt is creating a response. And even in rainwater, they are very sensitive to change in salinity of the soil. That's why they come out. And also, what else is there? Like I have another question that now the rain is consisting of some amount of acid also, nitric acid and hydrogen chloride, HCl. Acid rain. We are having acid rain everywhere. So is it also an irritant, or do you get a response that way also?
Ma'am, we have not tested that, like we have not seen using acids, but we have done with NaCl. But there can be one more reason that during rains, the water level is very high in soil. Like there is a lot of water, and earthworms can't stay in water for a long time. So that is why I think they escape out of soil, and we can see them climbing walls.
Has oxygen anything to do with it? Do you think once the air spaces are clogged with water,
Yes,
there is
ma'am.
no oxygen for earthworm? So
Yes, ma'am.
could that be a reason why
yes,
they come
that
out?
can be a reason, ma'am. Because they breathe through their skin. So
Right.
due to much water, they I think they can't get oxygen, and that is why they come out where they can get more oxygen.
So you mean to say that air, the oxygen of the air, they can easily breathe, but not from the water in the soil?
Yes, ma'am.
Yes, ma'am. That's what I think.
Can we also say that? Can we also say that they come out of the soil due to like for the migration purposes? Because if the rainwater falls on the soil, it clogs the surrounding, so it becomes difficult for the earthworm to travel. Rather than it will prefer moving on the surface and traveling, because considering there are no other.
Yes, Vivas. Actually, that is one reason that earthworms come out because they need a moist space to move on. They can't live in watery locked spaces in the soil. So they can come out to migrate from one place to another, and also for mating.
I can also see during the flooded times, due to the excess of water above the land also, would be water remain there. So I can see many earthworms are died. So what is that because of water?
Lakshmi, I didn't got your question. I didn't get your question.
Uh, I was asking that during the time of flood, water is always remain above the land. So I can see many earthworms are died during the time of flood.
Yeah, we can say as we talked that earthworms can't live in water. They will always die if they don't get enough oxygen to breathe.
But water has oxygen, na? So they should be
Uh,
able to even survive in just plain water. Or
Um, Ma'am,
No, no,
there's
very
one more
little.
where
Water
there's
has very little dissolved oxygen.
yes, and there is one more factor which I think because earthworm has a slimy mucus layer on their body, and if
Mhm.
there is a lot of water around them, there is a possibility that it can be washed off, and they won't be able to breathe because the oxygen will be not, it will be not passed in their body.
Yeah, so maybe you can try out this, by taking earthworms and just keeping them in just plain water, rainwater, and see how many hours, how many days they may survive. So I mean, you'll
Actually,
definitely.
ma'am, we had I had tried to keep them in water, and
With
they
water,
eventually died
plain
in
water.
almost yeah in clean water, in fresh water from the
Okay.
tap water. I took the tap water,
Ah.
and they had died in almost 5 hours, I think, almost five
Okay.
hours.
And
So
even if
we
we
can
keep them in
calculate.
water, what are we going to feed them?
Huh.
So that is why we are using cup culture method.
Okay. Yeah. So there was some interesting question on YouTube. I mean, suppose if you're, somebody said home cultured earthworms. You're doing all this culturing, creating cup culture in home lab. So what will happen to those earthworms once you release them into the natural soil? How would they get adjusted, or would there be some adverse effect? And I think it's just a question that you can think by thought experiment. If you haven't done that, what would happen?
Ma'am, we haven't done that, but what I think is they will able to survive because when we bring native earthworms normally from soil and we keep them in tissue paper,
Mm.
they eat that tissue paper and they start surviving there. So I think again, if we like put them back into soil, they will again start for survival. They'll have to eat something, so they'll start eating that soil, and I think they can live there.
So you are
And
saying
also,
they'll
to
adjust and they'll
Yeah.
not throw any tantrums. No, I don't want soil or something like that.
Yeah.
And also adding to Ritik, I think that also depends on like how much time we are keeping the earthworm. If we are keeping the earthworm for long time, like maybe for 3 months or 4 months in the cup culture, and then we are transferring it to the soil, maybe it can face some problems out there.
Okay, I think we have I think 5 more minutes. Okay, I mean maybe 3 4 minutes. We can wind up if we want to take some important questions. We
Ritik,
can
can
take.
you ask me one question, Ritik?
Yes, ma'am.
Ritik, in your experiment, after adding your NaCl concentration, at what time it will start dying? Actually, you haven't mentioned that one, no?
Ma'am, I didn't get your
Timing,
question.
whether you check.
Hello.
Hello, can you hear me?
Yes, ma'am. Now I can hear you.
Yeah, in your experiment, after adding your NaCl concentration, at what time it will start dying, the earthworms?
Ma'am, we actually we are not looking at the time when it is dying. We are trying to find out. So in that gustatory response assay, uh, we are seeing where the earthworm goes, if it repels from the salt filter paper, and in RER we are expecting that it will escape out of the test tube. Uh, for like in 30 seconds, I think the earthworm should escape out of the test tube in RER. but
Okay,
we are
fine,
not
fine, Ritik.
expecting it to die.
Okay, fine, fine. Ritik.
Okay, so maybe, I mean, we will just wind up this presentation, but while talking about this rainy thing and the rainwater and pH and millimolar and all, so maybe next time, see, this rainwater is also collected by the Pagalapos group. I mean, for their, they have another interest, their objective is different. Maybe of that rainwater itself, the earthworm group also can measure. I mean, Vaibhav, you were saying ki maybe that rainwater is of some concentration. So, is there possibility that you can measure the concentration? I mean, think about it. It is just a just a thought to design an experiment. Okay, so this is your last slide. So maybe we can keep this slide for say 1 or 2 minutes, and then we can wind up this presentation.
So as we can see in this slide, that yes, we are using earthworms for basically right now for ventral nerve cord regeneration and gustatory, but our seniors did amputation on them, and we are also doing earthworm culture. But through these basic things in our organism, we are able to understand so many other topics, like study of the nervous system, as we can see over here. Then we, in earthworm culture, we know that there is a tissue cup culture method. There is one vermicomposting. Then we are in the end able to understand our curriculum. Like, for example, cell biology, from epimorphosis, from regeneration, that we undergo, the cell undergoes differentiation, cell division. We understand how cell biology is coming in place there. And in gustatory response, how the earthworm is making decisions as to whether it wants to go towards the water side or the salt side. It decides its own environment, and there the chemoreceptors are responsible for that mainly. There also we are able to study cell biology, and there are many topics that you can study through this model organism. And home culturing is a very easy thing. If you guys need any help, we are able to explain it again, and we can continue this group discussion. And any doubts you guys have, we will clear them out.
Vaibhavi, what is the
Yes,
conclusion of your study?
Ma'am, in the end, earthworm can be used as a model organism for many researches. So till now, earthworm has not been as exploited as we see Drosophila and nematodes. But yes, earthworms can be used as a model organism in many researches. Once, once in one chatShaala, we even thought that we can use earthworm for fibrosis, cystic fibrosis. As we know about the breathing that it takes place through its mucosal layer. So if that mucus layer dries, so that earthworm won't be able to breathe, it will suffocate. Similarly happens in cystic fibrosis in humans. If something like that happens to us and the mucus in our body gets dried up, so we will end up having difficulty in breathing. The same way we can collaborate and understand the earthworm in a way and use it as a model organism for other diseases also. So we want to exploit the earthworm more and more because there are many similarities. For example, the earthworm also has CNS, PNS, that is central nervous system and peripheral nervous system, like us humans. And it even has a similar circulatory system. Like we have 4 chambers in our heart. The earthworm has 5 arches and in a closed environment. So like it's like a tube. Like we have our veins, arteries. Similarly, the earthworms also has those. And the main thing that I am studying in earthworm, that is ventral nerve cord regeneration. Nowadays, we know that paralysis, when it takes place in humans, so the human cannot regenerate back its nerve cord or its spinal cord. But I have seen in earthworm that in 48 days, the earthworm regenerates back its nerve cord. We have done this, and we are going to do more experiments under this research question. So this is a thing that this will help the mankind or the society, even if it is only 1 or 2% contribution to the field of regeneration. This will help the and we can use earthworm as a model organism. That's what I meant. That was our conclusion of our research.
Okay, thanks. Sorry, due to the time restrictions, we will thank the Earthworm group, and please post your questions in YouTube as well as here, BBB. It will be recorded and shared in the groups. And we now move on to the group on Hydra as model organism. Are all the members of the Hydra group present here?
Yeah.
Okay.
Manmasi, please add the audio. Yes.
Culturing Hydra is very easy. Take approximately 80 ml of dechlorinated water in a 100 ml beaker. With the help of a dropper, we transfer the hydras in that beaker. amputate hydra, we place the hydra by using a dropper onto a slide. We place the slide under a microscope and then, using a blade, we amputate the hydra into two pieces.
This video is really nice.
Yeah, nice. So can we have the earthworm group unmuted, please? So there are some questions already pouring up.
I think everyone's doubt is Hydra is a plant or animal.
Okay, that's a big question.
Yeah,
Why did
everyone
you
is
have that question?
confused.
Yeah, but why did you have that doubt first in the first place?
Yeah, because when we see this Hydra, we can see it is green in color. So anyone from this Hydra could explain that?
So
अरुणिमा अच्छा
yeah,
विवास is taking ठीक है
okay,
ठीक है
yeah,
ठीक है
I'm
carry
taking.
on.
Yeah, so Arunima, we talk about anything we see green in color refers to, or like simply talk about plant. So can we know that why is the green color, or which is the thing that is giving is it's the green color in case it is plant? So can anyone tell like what is the green color is for?
Is that chlorella?
Nay, normally in plants, what is this green color for?
Chlorophyll.
It's the chlorophyll.
Chlorophyll.
Okay, so chlorophyll, is it chlorophyll present in the animal?
I don't think Hydra has chlorophyll because I have seen hydras of other colors also. They are white and
Yes.
brown also.
Yes. So what happens in the case of green hydra is it remain in a like it resides. It gives space for other bacteria to grow along with it, which is chlorella, which gives a green color to the hydra. So we tell it is a green hydra.
What is chlorella? Which kingdom or which group it belongs? What do you mean by chlorella? This is we are trying to make Arunima, Anamika understand.
And Bivas, is it a hydra or it is an algae?
So chlorella, I think, is an algae.
Algae, yeah.
Yeah.
And
it is
what type
living-
of? Yeah, adding to your, please just explain that this association in which an organism is living inside another organism, what type of a condition it is? Like the first question, like Arunima said, that whether it is an animal or a plant because of the presence of green color. So you gave an example of maybe a bacteria is present. So this kind of association, what is this kind of association when an organism is staying with another organism? Can you please explain?
Is that symbiotic association?
It's a symbiotic because both of the parties are getting benefited from this.
Okay.
Because the chlorella is getting shelter and the hydra, even if there is no food, chlorella can produce its own food through photosynthesis and it can give it to hydra.
Okay, okay. So it is residing inside an animal's body and providing it with food in adverse condition. Is that so?
Yes, and even the hydra, if it gets some food from the moinas, it surely helps the chlorella and also gives it some shelter.
Yeah.
Okay. So it eats only moinas?
No, it can eat any water fleas, I guess.
Okay.
But why did you feed them with moina?
Because both Moina and Hydra are freshwater organisms. Like Hydra is also found in pond and like pond lakes. stuff, and even Moina is found. So it's kind of an ecosystem building up.
So,
The Hydra, it's Moina.
yeah, this question that whether Hydra is a plant or an animal, Arunima, is it clear? Like it is catching food, so it is an autotroph or an heterotroph? It is making its own food or it is catching food. So what is the characteristic?
So ma'am, the chlorella that is there on the hydra, that is autotroph, whereas the hydra itself is a heterotroph.
Okay.
But considering what Hina ma'am is saying, if we only differentiate between plants and animals being autotroph and heterotroph, there are many plants which are heterotroph as well. So
Yes,
yeah,
yes.
so can we
Insectivorous
say
plants.
this as a distinguishing characteristic between animal and plant?
For example, Venus flytrap.
Pitcher,
It's like
plant as
pitcher
well.
plant.
Yes. So it captures insects, right? The pitcher plant.
Yes.
So what is that relation?
I think both are insectivorous plants.
Okay. So, but what do you call them? Symbiotic, parasitic? I mean, what is it? Another one, commensalism.
We can call it parasitic or even saprophyte. No parasitic, barabar. We can call it parasitic kind of plants.
Mm-hmm.
They live
Because
over the other plant, other food prey for their own nutrition.
But they don't. But why are you calling it parasitic? Like, why can't we call it heterotrophic?
Because all parasites are heterotrophs or all heterotrophs are parasite. Which statement is correct? I think you will get your answer. Heterotrophs can be.
All heterotrophs are not parasitic.
Yeah.
Which
But
is the
all
correct
parasites
statement?
are heterotrophic.
Yes, that is more correct. Yeah.
Yes.
That means heterotrophs can be holozoic. Like Hydra is a holozoic animal, isn't it? It is catching its food and it is engulfing it. And along with that, it is showing symbiotic relationship. So how is it an animal? I think we need to clear it out before we move forward, because that was the classification which was showing up in the slide, and we didn't catch up with it. That's why this question has come. So I think, why is it an animal? We have to justify this answer before moving ahead. Why does Hydra belong to kingdom Animalia? Anybody from the Bivas?
Ma'am, Hydra has a tube-like body, as we are seeing in the photo, and it is engulfing other organisms for its nutrition. Okay, and it is metabolizing that food. So, and it has tentacles like stuff which can kill the other organisms, or at least paralyze them. Like it can paralyze the moina. So we can call them animals because even Hydra shows budding. And in budding, it also gives rise to another organism. And if you cut an Hydra, if you cut an Hydra from any part of its body, it will regenerate back.
Okay.
So I guess these are some characteristics that will help us tell that an Hydra is an animal. And it comes under the classification.
How to you can say, like, if we like, how can regeneration be a like different factor between
point of,
animal and plant? Because plants can also regenerate some part of its own.
So the basic, if we go to the basic classification, animals are heterotrophs, as in I'm going on very basic level in school what we've studied, and Hydra is a heterotroph. So I guess that that is the answer. That is why it is placed on the kingdom animal.
So can we say Venus flytrap was also animal?
It's a parasitic plant. You got your answer.
Then again back question to like what Hina ma'am asked. Like what is the difference between parasitism and heterotrophic?
So, looking to the classification, it belongs to the kingdom Animalia, and those who belong to Animalia, they have multicellular. These are eukaryotic organisms. So,
the plants
Hydra
are
is
unicellular, Rachel.
No, see, we can resolve this question just by the mode of nutrition. So Venus
What about
flytrap.
cell wall? Cell walls?
Yes, go ahead.
So I would like to say something. So every heterotrophic organism, so an organism which depends on others for its food is called a heterotroph. Okay, and an organism which feeds on other organisms by deriving out nutrients from other animals and making them their hosts are called parasites. So now we can say that every parasite is a heterotroph, but not every heterotroph is a parasite. Because we are not living on another organism and we are sucking out nutrients from it. We might be eating other animals, we are not sucking out any nutrients like in that way, like in a parasitic form.
So does Hydra have a cell wall in its cell? I think basic answer I was looking for.
What Chitra ma'am was pointing out, that plants have cell wall.
Why don't we talk about basics? Because small children, school children are also there, and that was a question which came from here. That what is an animal? How do we say Hydra is an animal? So now can you just please explain? Is there cell wall in it? Is there a vacuole present in it? A big,
And
small
also
food
we
vacuole?
can talk about plastid as a whole organelle. Is it present in the animal? Because that was the first point I brought up when we talk about why the Hydra is green and why Hydra is green, because
So,
chlorophyll
Bivas,
is
that
present
answer
in
we have already got, that it is in symbiotic relationship, and it has inside the cell wall, inside the cell membrane, the body wall, there are present certain algae which are giving the green color to it. That answer we have got. So but how it is a plant, how it is an animal, that points we were just trying to discuss.
Can I
And
say something?
Yes, please.
Is that not all
I
hydras
am
are
from school and I study in science that Hydra is a plant which is found in fresh water. So I said this is a plant. This is a plant.
So Anamika, can you tell me if plants locomote? Can plants locomote from one place to another?
No.
So I'll tell you a fact about Hydra is that Hydra locomotes. It can move from one place to another. So do you know what is somersault? As in a human does a somersault. Do you know that?
No, I am in only 7th class. So I
Okay.
really know
So there is this activity that humans do where they go 360 degree. The same thing Hydra does. Hydra uses its body to locomote from one place to another. So I don't think plants locomote, but Hydra does. So doesn't that make Hydra an animal?
So Vaibhavi, could you please add on what is the tentacle in Hydra? Please add on that.
Okay. So actually I don't know about details about the tentacles, but mainly the tentacles are used for two things. One is to kill its prey. So, for example, the Hydra wants to eat the Moina, so it releases some enzymes, and that paralyzes the moina. So it can eat
I
the
have a
moina.
question, Vaibhavi. Vaibhavi,
Yes,
I have
ma'am.
a question. You said that it releases certain enzymes. When we was looking at the classification in the slide, we saw that it belongs to Nidaria. So, how come this name Nidaria is related to some part of Hydra? The name Coelenterata is also related to its body wall. So, can you please tell something about the Nidaria? The name Nidaria is coming in the classification. Can you please show that slide of classification? That is a very important thing because we are already stuck with animal and plant and yes. So here it was written Nidaria. Can anybody from the group tell us why this
Because
Nidaria term is coming? Yes,
the
please.
Nidaria term is coming from the special type of cells which are called nidocytes that we have studied. So nidocyte is basically a type of small injection for cells which inject the toxin to the prey, and the prey gets paralyzed. So that for that reason get the name it get classified under the Nidarian group.
Yeah. Okay. So they are stinging cells which sting the prey and then it engulfs the prey.
Taking up some YouTube questions like, what is your research question? Why are you taking Hydra as a model system?
So actually Hydra shows morphollaxis. So now what do we mean by morphollaxis? Is that if you cut the Hydra in any direction, for example, if you cut the Hydra, can you show a photo of Hydra? Yeah. So as we can see, oh, last slide, yeah. So as we can see, the Hydra is first cut longitudinally. Can somebody give me the pen? Okay. So as we can see here, okay, so the Hydra is cut longitudinally, but still the Hydra regenerates. And here the Hydra is cut in transection, as in it's cut from as in you can understand like how the pieces are made over here. 1, 2, 3. But still these 3 pieces give us back a Hydra. 3 individual Hydras. So this is showing that the Hydra has the power of regeneration. And it is said that the Hydra is an immortal organism, that it can never die. Even a
So
small
Vaibhavi,
part. Yes, yes, Kiran.
so the this part is interesting. So that looks like you have disintegrated these cells, all the part of the Hydra, and then again they combine themselves, and then again we see a body. So but I was expecting, as you can say, that each part can regenerate. So I was expecting that each part from here should regenerate and makes its own body.
Um, so see, what it is depicted here is, so they for example, this Hydra is cut into this part, okay? So now let's say the cells that are there in this head are these cells, okay? Now these cells get differentiated, sorry, they get undifferentiated and form stem cells, okay? So we previously know that they were differentiated cells of particular uh, metabolism that they had in them. But after the amputation, now they became undifferentiated set of cells. Okay, now these set of cells will undergo differentiation and they will form the other parts of the Hydra. Like for example, we have the head, okay, which can differentiate into any type of cells. For example, I am a human and I have been amputated, okay, and my lower body is not, which are undifferentiated and they can regenerate back my legs.
So we have Jude from Makerspace. He wants to ask some questions. Yes, Jude.
Yes, yes.
So that means this is a model system for regeneration.
Yes,
Because
ma'am.
that cannot be studied with other model systems. So this model system is to study regeneration particularly. Is that
Yes.
right?
Yes, just like
What about
Earthworm.
symbiosis? Vaibhavi,
Um,
besides regeneration, what about Hydra as the
We can study that also in Hydra. Like we
Just a
saw
minute,
in
Vaibhavi, just a minute. Sorry to interrupt. I am told that some group members are here, so I would like them to unmute and then participate in the discussion. I think Rachel and Tulika, right? And Abhijit Vinod, you are part of the Hydra group, right? Please unmute yourself. And there was in video, it was shown somebody is doing amputation. Please take over and yeah, before that, just before these people unmute, and then Jude, do you want to ask your question? Please go ahead, unmute and ask your question, please, Jude.
So my question was probably already answered now. It was about why is that Corollera not a parasite and considered to be a symbiont? And the second question was, why does the Hydra not always have that?
Right. There are some Hydra which don't have chlorella, the brown Hydra, and other varieties also. So that may also help to clear out the confusion whether it's an animal or what you say, plant. Not all Hydra are green, and not all green-looking creatures are always plants. So, are the other members of the Hydra group present? Please unmute and take part in the discussion and explain the experiment.
So
We have 15 minutes to go.
Yes, Rachel.
Yes. To study regeneration in Hydra. Vaibhavi was also telling that it shows the morphal axis. It means that if we cut its part, so it will regenerate because it is having the stem cells, so that it can regenerate. So the application of this regeneration is that to study how we in humans the healing, and yes, Hydra also shares some genomic sequence with humans, like this Alzheimer's that the genomic sequence of Alzheimer's that also found in Hydra, so that it can also be studied. Yes.
Other members, do you want to add, and or do you want to show the experiment? By the way, is just morphal axis one type of regeneration? Aren't there any other types?
Yes, I wanted to ask a question like, is there any difference between the regeneration of Hydra and regeneration of Earthworm?
Okay.
So I think yes, is because if you cut earthworm in two pieces, only one part will regenerate. This experiment was done by our seniors, and if you cut Hydra in three pieces, here the demonstration is given. All three pieces would be forming. So if you cut the Hydra in three pieces, you will get three different Hydras. Each part is generating the whole Hydra.
So it doesn't happen in earthworm that way, yeah?
Yeah, so if you cut earthworm in two pieces, only one part will regenerate. And I think Vaibhavi is asking one of the major questions relating to that. Yeah.
So can you consider the cells as totipotent cells of Hydra, which have the ability to regenerate any part?
Yes,
Which
ma'am.
cells are totipotent?
We
Yeah.
can conclude from earthworm and Hydra is like in Hydra, the totipotent cells are distributed throughout the body. So if we cut at any section, we are getting the whole Hydra generated. Whereas we can conclude this as well in earthworm, the totipotent cells are localized in one of the parts.
Which cells
So
are
here,
playing the role of totipotency in Hydra?
Leaving the tentacles, the whole body of the
Body is
Hydra has
totipotent.
totipotent cells.
Yes, I do.
Leaving the tentacles.
so if we
Yes,
cut
ma'am.
the tentacles, if I go about saying, if we cut the tentacles into 2 or 3 parts, the tentacle will not regenerate.
Okay.
So, Saida, in this experiment, we can see the regeneration.
So Hydra can, as we are also saying that it is immortal, because this is what we say, because it is renewing itself. So uh, it may grow around 5 mm to around 5 mm, as it is having, because it contract itself and then it expand itself. So yes, it can grow around 5 mm.
Okay. So Rachel, Anamika is a 7th standard student. Still now she is confusing with what is Hydra and where we can see. Could you explain a little more to Anamika?
Yes, please.
Yeah, please.
Yes, Anamika. So
Both are
this is,
both are classmates.
And they are very much interested to this Hydra.
yes. So these Hydra, you already know that these are found in fresh water. So when earlier, when this is my experience, that when I was looking for Moina in my locality, I was collecting water samples from different ponds. So I got Hydra from there. So yes, we can find mm, in the ponds and lakes, because it is a freshwater animal. Yes.
Rachel, can you tell how you developed this home lab for Hydra?
So actually, I had Hydra, but few months back I lost, because I didn't have much Moina to feed them, and
That's
maybe
okay.
there is another goof up. So,
No, that's
but
okay.
yes,
Yeah, you can tell
yes,
what
but
kind of media you used, or it just water. What kind of water? So I mean, Arunima is interested, na? Arunima or Anamika. So
so to culture Hydra,
yeah.
we use dechlorinated water, and then we transfer Hydra in this, and we feed them crustacean, as we are already having Moina with us, because we already cube introduced Moina, so it feeds on
हाइड्रा
these
कहाँ
crustaceans.
पाया जाता है?
Abhijit,
Fresh
Abhijit
water.
of this group, can you unmute and please help your friend Abhijit Vinod? This is your picture, na? Green Hydra by Abhijit Vinod. So please
Yes,
help.
ma'am.
Yeah.
So regarding the culturing, I actually got the culture same as Rachel. I also got them from a pond near my college. And what I did to culture, like to collect them, was I took a bunch of water plants. That is, particularly saying, I took hydrilla plants and I kept them in a bottle. hydrilla plants along with some water, and I kept them in a bottle overnight. And this picture is actually from that same bottle actually. You can, and when kept overnight, the hydra actually, and when observing the next day morning, these hydras were seen found in the walls of the container of the bottle in a very abundant number. So they came out at night and came and stuck on the walls of the container. That's how actually I got the hydras. And in the same way, when I took that model, I took that in the concept the same way, and I started culturing them in my house, in my home lab, my house. What I did was I separated some of the hydras and I added a bunch of plants and used dechlorinated water, and I kept them near the window side. So because they have the chlorella, because they are green hydra, they have chlorella, and they can use the sunlight as a like what to say, chlorella does provide them nutrition. Therefore, I kept them near the window sill to get the sunlight, and they would survive. Even if I did not feed them all the time, they were able to survive like that. That's how I actually developed that home lab of hydra. Actually,
So, Abhijit, if we will not feed them, it can grow.
yes, they can actually.
What do you mean by growing, Arunima? Growing in size, length, or multiplying?
Yeah,
What do you mean by
yeah.
grow?
If cell can it can grow by height and size.
I think they can survive without food. I don't think anybody has done the experiment.
I did an experiment on that. Hello.
Yes,
Arunima, I did an experiment on this topic of effect of starvation on green hydra. Because they have got the symbiont, then they can not just grow in height and body, they will also reproduce. They carry on with asexual reproduction even during starvation, and for at least 7 weeks they will survive without any feed.
Yeah, Batul ma'am, I think it's very interesting. They can grow without food.
They can survive
Yes.
without.
Yeah.
Why? Only green hydra will survive? What about brown
Yes, because
hydra?
brown hydra wouldn't survive because I don't think it has any symbiotic association with chlorella.
So brown hydra is having just like green hydra. Green hydra is having chlorella. So just like green hydra, brown hydra is having another algae. It's vulgaris, if I'm correct. So
That's amazing. That each and every hydra has a symbiotic association with another organism.
Yes.
So Rachel, what are the foods we can give to hydra?
Are you sure,
other
Rachel?
than-
Are you sure, Rachel, that brown hydra has got vulgaris?
Yes. So this is because of this symbiote. Its name also suggests that Hydra vulgaris.
Or is it that green hydra has various symbionts?
No, in that case, shouldn't brown hydra be green in color? Algae are green.
So if I was to hypothesize, I would say that the green hydra would survive in starvation, but the brown hydra wouldn't. But according to Rachel, there is a symbiotic association with some other algae. So yeah, we can try finding out that.
Yes, because we already saw that green hydra is surviving for a long time by Batul, but we haven't seen that brown hydra is surviving for so long without food. So we can see that.
Yeah, and how does hydra reproduce? I mean, we don't usually, amputate hydra to see that it grows into many numbers. Naturally, how does it reproduce and grows in many
So
numbers?
naturally, hydra in normal condition, like it grows asexually by budding, and in autumn it 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 reproduce sexually, because it also sorry.
Do you want to show the budding wala hydra pic if you have from your home lab? Hello, what happened? Hello? Okay.
They are having azan over there. Maybe that's why.
Okay. I think the time is also up. The Hydra model organism has actually sparked a very interesting, very basic discussion, I must say, whether it's a plant and animal, and we need to check what are the characteristics that makes something an animal, what makes something a plant. So that's why a school student is interested in this as a model organism, and even to develop it in the home lab. So I think we'll stop this presentation. There are questions pouring out even
Ma'am,
in the
please
YouTube
last
channel.
question.
Sorry. Yes, Anamika.
Ma'am, can Hydra have any bloods in their
Blood.
body?
Oh, can it have blood in that body? So when we cut open it, we can see for ourselves, right? Whether it gives out blood, right? So if you want, if you are guessing that it will have blood, then it should look red, right? Because blood is red usually, right?
But ma'am, in insect there is white blood, not red blood.
Oh, so why do you think it's a blood?
Okay, ma'am, I'll take it up.
Yeah.
Actually, Hydra is a non-chordate. It doesn't have a backbone like we have, okay? And it doesn't have a heart and a blood vascular system. So it doesn't have blood. It is a invertebrate. उसके backbone नहीं होता है. तो वो lower animal है, lower level का animal में backbone नहीं होता है. So it doesn't need, because it is a marine animal, वो पानी में रहता है, उसके अंदर body fluids blood के जैसे नहीं है. लेकिन normal, but this is a very good question, that whether they have body fluids or not. I think the child is asking blood, but it's body fluids तो होगा, लेकिन blood नहीं होगा.
So let me finish that how Hydra reproduce naturally. So I was telling earlier.
No, bloody fluids क्यों चाहिए? The question is, why do we need blood? Right? That is to give oxygen. But does Hydra need oxygen? That's the question.
Yes, ma'am. Hydra is already getting sufficient oxygen, and there is all chlorella inside it. So, should I continue telling, or?
Yeah. Why don't you just wind up what you were saying?
So I was saying that in natural condition, Hydra grow by budding, but in autumn, so autumn is a cold season. So in this condition, Hydra is a hermaphrodite, so it also produced by sexually. It means it grows also by sexually reproduction. Yes.
I mean, it will be better if we see some budding hydra pictures from your cube home lab, just to show, I mean, how it grows. No, not this schematic. I meant if you have from your gallery. Yeah, I think there's something you can see as a bud. Or anyway, we'll call it this as an end of the presentation. Further discussion will continue on various groups. So I think we'll have a short break now, and I'll pass over to DP now.
Yeah.
Thanks.
Thank you, Meena, and all the groups, of all the members of Earthworm and Hydra group for showing us what you have been doing for all these days. And thanks a lot for participants who have joined on YouTube as well as here and engaging in interesting conversations, making comments and asking questions. So as Meena just said, we are going to have a break from what we have been seeing. And in this break, like, we are going to have some surprising things. And I would request Arunan to introduce and talk about it as I upload the presentation regarding the same. Over to Arunan. Is Arunan here? Yeah, so I think the name is out there. So this is, yes, Arunan, please.
Yeah, can you, okay, can you hear me?
Yes,
Am I
yes,
audible?
we can hear you.
Okay, so this is usual awards, annual National Meet Awards. We have a long list as usual, and this time, of course, it is much more thanks to the home labs across the country. So, can you hear me, DP?
Yes,
so
yes.
can you just roll the screen, that list?
Sure.
Maybe slowly, and we can go to. Okay.
Yeah, I have them as slides.
Oh, you have them as slides. Okay.
Yes.
Okay. So just move one after the other, and then we will come back.
Okay. Sure.
So interestingly, I think almost countless awards. When I counted last, it was 91 awards under 9 categories. So these are the certificates that we give out every year. This time, of course, we have made certificates, but it will be released later. So DP, can we go to the 5th item?
This is the one.
Possible?
Yes, this is the one.
Okay, so so why I took the 5th one, the first to 4th I did not talk about it first. I didn't want to talk about it first, is because this is where the real happenings occur. So outreach. So Anil Sadgopal Award for Popularization of Science and Contribution to Science Outreach. This outreach we have is outreach for cube-based studies and associated studies. So these people are the people who contribute a lot by connecting with other people. Now I can just take a few examples because so many names are there. I can take a few examples. Sham, probably the most of the work that is happening in Thrissur, Kozhikode, Coimbatore, etc., has been initiated by just one person 2-3 years back. That is Sham, who happened to be in Mumbai long time back, and he is running a small sort of an firm in Palakkad. So he went back to his college at I think Thrissur. This is SN Natika College, and through that he sort of mobilized a large number of people and centers, including centers in Coimbatore and Kozhikode and places like that, which we would never have been able to do that. Now, I'm just taking Shyam's case as one of the classic cases, and likewise everybody's. For example, Ganesh from Ranchi has been, doing enormous amount of outreach work, and Sarita recently. So I think all these people needs a lot of discussion, but I think we will not go into that. Sarita recently organized a big Jamboree session in Delhi. Chitra has been one of the stalwarts. Sudhir Verma has been also. So many people. So I just don't want to go to all this. Other than that, the Northeast, we have Krishnendu has been doing this outreach program there, and Sushanta. So that's the point I wanted to say. Several other people. I think the name. I think we will read it out later. So next one, so this is Anil Sadgopal Award, and the reason Anil Sadgopal was one of the first to start a major science outreach program from TIF for a long time back, I think 50 years ago. I think we are going to celebrate that, the Hoshangabad science teaching program that started 50 years ago in November. So next slide, please. We will read out this a little later after. Okay, K.S. Krishnan Student Mentor Award. Now, without student mentors, I think this will never happen. Now, how much the teachers are doing it. Now, this is only a few people that we have highlighted now. I think there are so many people, student mentors. Now, this could be a student mentor just 2 months after I think a person can become a mentor, maybe 1 month after she or he can become a mentor. Maybe even after a week, a person can become a mentor. So we have plenty of people like that. So Deepika Iyengar. So the names are not just because somebody merits more than the other. Each one is a great contributor in order to build up this huge network that we have right now. So you can see that even 91 people that we had to sort of highlight now among a whole lot of people, somewhere around 1,500 or more than that, itself is a big job. Rachel, you must have been hearing Rachel. I think all these people you have heard in the last 2 days as well as today and in the coming sessions. Omkar, Nidhi from 11th standard, 12th standard student from Goa. She has been one of the stalwarts now. Shalu from Patna, Anshu and several other people. Garima, Delhi. Okay, I think I will read out now Komal Kotra from Delhi, Pulkit from Delhi, Kunal Ameya, Anjali, Kunal Ameya from Mumbai, Anjali from Chennai. Consistently for the last 3-4 years, I think she has been doing work there and outreach work. Abhishek Unikrishnan, Sujit, new entrant, and he has been contributing a lot. Manisha Rani from Ranchi, I think we heard her yesterday. So Oindril, I don't know whether Oindril is here. Asansol, one of the areas in West Bengal, I think Asansol has been represented by him and he has been doing some work there. Kanishka Parashar. So we have plenty of people. Deepika Iyengar, Rachel, Mansi from Ranchi, Manju and Jojo. Yeah, okay. Okay, so next slide, please. This is K.S. Krishnan Student Mentor Award. Now, some of these people are stalwarts, a huge contribution they made in science in various areas from TIFR, some of them. This is another TIFR stalwart, Veronica Rodriguez. Unfortunately, she's no more. The award is given for the perseverance in pursuit of science. And this is an enormously interesting thing because without these 7, 8, 9 people, I think we probably would not have been able to manage this much of activity in the last lockdown period. If you look at the chatShaala, I think you can see their frequency of their discussions and contributions in writing has been enormous. So Arunima from Kozhikode, Ashwati from Natika, Siddhi also is from the same place, and Lakshmi from the same place. Abhijit, you just heard him his escapades with different type of organisms that he got it from the ponds and rivers from his place. Shraddha from Mumbai, she now is in, I think currently she is in some area in Maharashtra, but because she went during the lockdown. From there she works. Nasish contributed, and you heard her contribution on the when in collaborating with the Ranchi group on cardamine. Vaibhavi just heard, I think you have been seeing them. Yes, you have been hearing and seeing him all the while. Ritik is here today just now. So these people, their perseverance for the last many years is what, and some of them of course recently have been the real contribution that we have in order to get to this sort of things happening. Next slide please, DP. Now I will be carried away by talking about Veronica Rodriguez or Didi Kosambi, which we will refrain from. Didi Kosambi, I think I will have to say something about it. Didi Kosambi is a mathematician, was a mathematician in TFR, statistician in TFR, but he contributed immensely in biology, particularly in population genetics. Anyway, so Didi Kosambi was also a historian, a lot of history books. So I think he's a polyglot, polymath. So this award is a very interesting award because this is the Young Scientist Award. All the up and coming people, and some of them who have been there with us for some time, I think who have been not been probably given award earlier, is picked up for this, and they have been contributing a lot. I think they are the real field workers. So Shipra from Ranchi, I think this year she joined. I think she's from Patna, but she's now in Damkal, I think. Patna Women's College. Anjani Kumar from Mumbai, Mansi Prasad from Mumbai. Mansi has been there, but I think she has been highlighted this time again. Tulika Sharma and Kaninika from St. Xavier's College, I think they just joined and they have contributed immensely, and we are waiting for them to really show that they will be the ones that will be carrying the flag forward in St. Xavier's College particularly. Of course, Bivas is there. Well, I think his name will be coming somewhere else. Kaninika Akansha from Ratnam College, I think reasonably silently she has been doing, but she has been contributing a lot. And Aditya Joshi, Ulas Nagar, he is I think he has another name called Om Shanti. I think that he is known as Om Shanti everywhere in the writing. Rahul is a school student from Kanpur. He has been, these are all field workers and they have been contributing immensely. Deepak joined recently from Ranchi and he has been contributing immensely in the last few months. Tejashri, this is another very interesting exploit that we have from the Andhra Pradesh area. Nellore and Visakhapatnam areas, I think we are getting a lot of people, and these people are doing immensely wonderful work. Farhan, Indore, we have a small number of people who are through the maker space.
Yes.
And Prashant is a I think, I am not very sure, he is a physicist. I think he is recommended through one of the principals of he was a principal of Bhivanti College. Later, I think he shifted to Bandodkar College, and he is one of the cubists. Moses, and he recommended Prashant, and Prashant came, I think, in 2 weeks ago or something, maybe a week ago, and he has been, I think, one of the real field worker for us now, right now, and in all areas, including biology and physics and chemistry. So Sri Lakshmi from Cochin College, and these are people who joined recently, but we are having high hopes on them. Alina from Cochin College. Cochin College has got, I think we will come to that a little later probably. Cochin College has been with us for quite some time, and they also have got extension programs with the schools. So some of them are school students here, and the next few people, you can see nature mechanics. Hishma Biju is, so the nature mechanics is a small group of mothers, grandmothers, I'd say all women group. The daughters, mothers, and grandmothers, and they have been, I think yesterday or day before, you have seen some of the contributions of them while discussing about the cardamine studies, seed germination studies. They have done extremely wonderful work there, and they have been contributing immensely on mango mapping and all other things. Amrita Lakshmi from again Kochi, that's our school extensions that we have. Then Muhammad Hanim and Rokia, these Muhammad Hanim from Kochi and Rokia and Muniba from Kirurimal College. Kirurimal College has been rather silent, but I think they have been very active now. So DP, next one. I'm sorry that I'm taking a lot of time. I think unfortunately we don't have any chance for clapping, is it? When we are announcing the name, otherwise there will be a lot of clapping.
They can
Going on in the chat box.
Oh, you can. Yeah, chat box and all that. You should have that. Okay. Okay, so SK Mahajan Award. Okay, SK Mahajan Award for the popularization. See, essentially everything is popularization of scientific pursuit. Everything is making it popularizing science and scientific pursuit, science education. That's what our main theme is. And SK Mahajan has contributed a lot, both in his high-level work in DARC, in molecular biology, and also in propagating science and scientific temper when he was alive. I think, unfortunately, he also is no more. So here, this popularization and scientific pursuit is popularization of scientific pursuit is a very, very important aspect. So I was mentioning about, I will not mention about Vivastag. You have been hearing him, how he is popularizing, and I think the latest thing is that I think his brother's school is being roped in Delhi or Faridabad. I am not very sure. The other is the next names are Sulekha, Sudhagar, and these are again the nature mechanics people. The mother, grandmother, daughter gangs, so many of them. So Reggie Biju was the person I think who and Hishma, her daughter, where she is actually a medical student. Both of them did that seed germination studies, beautiful, very neat seed germination studies, which helped in for elucidating ideas on seed germination studies in cardamine. Shija, Shija is a school group. She has been contributing for the last 3 years, I think. And the mango mapping that we are getting to know that mango flowering starts in 8 degree latitude, that is near Kanyakumari, and Thiruvananthapuram has been actually initiated by Shija, and she has been the first one. From September onwards, she will be sending the mango flowering mango trees photos from there along with her students. Very, very ordinary school. School students, they managed to get the mobile camera photos and sending it. Now we have another connection that we got through the Nartika group, I suppose. Nizi, who is here, we just heard her, and then we are banking on her also for the connection in Coimbatore area, and she has already got enormous amount of exciting students with excitement on particularly vermiculture and stuff like that. So we are so these are the people actually whom we are banking on for continuity. Sanya Panda, I said about Visakhapatnam earlier, so I was showing that that was the area that was not very much covered, though we had some Gundur and Hyderabad, we had some centers which are not very well working, but Nellore and Visakhapatnam area, I think we are having. So Sanya and Sri Rajini are the people who are. And in the next one, I can't Manzoor Javaid, you are not hearing him so much because he is very active in other cases also, but he is going to organize a big campaign there in Jammu and Kashmir, and no, it's not Jammu and Kashmir, that area, whatever that you call it. And Ram Kumar, Puducherry is another center that, of course, we had a Puducherry center there earlier with the Kajal and Abhishek, and they shifted to some other place, and then Puducherry was almost drying up, and we are getting the school teacher group, in fact, through the mango mapping stuff. I think Rajkumar has been roped in by none other than Raghunath. Raghunath is one of the People Science Movement chaps in the Puducherry area, and he was. Now the Jaipur group, you probably may not be seeing of late, but I think we are having two new people. I think Sumita is not very new, but Somana Datta is starting now. So we have high hopes in seeing that it will be popularized in that area. And Bikaner, we have also some centers. So that next slide, I think we'll have to go back to, this is the last of the slides. We'll have to go back to the first slide onwards.
Yeah,
first to
I
fourth.
put them in the same order. So the next slide will be the
Okay. Okay, so then the first slide we need.
This is the one.
Okay. So this is the enormous contribution that we are hoping, and of course have been made by these two centers. So two centers have been selected this time. I think every year we have some centers. I think it's not a selection. Is not. I think is not the word we should be using. I think we just picked up. There are many, candidate centers. So two centers, SN College and Nartika, I think immense contribution they made, and we are hoping the major contribution right now happening in AND colleges, and AND colleges mobilizing the rest of the people in Delhi. And Kiro Rimal College, Deen Dayal Upadhyaya College, and of course Chitra's College. So we are hopeful that Sarita and Chitra, Sudhir and the group will be, and also Yamal and others will be organizing this through this. So next slide, please. Why Siddhi Award for Pursuit of Excellence to 2020? I think this is a very rare award that we handpicked and two people, and I hope that you will never blame us for picking up Syed of Elphinstone College. You have been hearing her for the last 2 days, and today also. Lydia Matthews of St. Xavier's College. She has been till now, I think till recently, she has been in Junjunwala College, and now she's in St. Xavier's College, I suppose, got her admission in Masters. And Lydia Matthews, you may not be hearing her very much, but she is the one behind a major organization that CUBE has. Every day's map, you get it that night at 12 o'clock or 1 o'clock, is because of Lydia. Lydia makes the map of people who have participated in each day's last more than 70 days of chatShaala. And so many other things. I think Lydia silently does that. So next slide, please. This is a slide. This is a sort of assistantship grant that we offer from Kishore Bharati Associated Organization. So this grant is given to Lydia and Drishtant for 2020. So that's a grant assistantship that we were giving it even earlier in some form or the other. So this is Lydia and Drishtant has been, and you can see that there is a contribution in several other ways. Okay, next slide, please. I think we are at the fourth slide.
This is the last. Yes.
Okay, this is the last. Okay, so the next slide, because I didn't complete that. Okay, no, this is the last. Okay, so okay, I think these names, if I read out, I think you will never have any dissonance. Ashutosh Mulle, yesterday you heard him, you saw him, and you have been hearing every day. And these are all stalwarts. I think Mayur was the first, probably Ashutosh was with us ever since we started. Mayur was as a student, he started with us. Rafiq has been there ever since we started again. Jay Kishan, you all must be knowing. Batul joined us lately, but her contribution is immense. So these are stalwarts. I think I should say that's from young stalwarts that we are waiting them to come up with ideas every day. And Kiran, I think we don't have to say about it. She's always here. Rahul, you may not be hearing him off late, but I think he's in Delhi and he will be coming back now. Lockdown period, he went to Delhi, and otherwise he has been with AND College, and from there he got into TIFR, and he's doing his PhD in TIFR, and right now, so he has been there with us, and he will be there with us. So that's the. So next slide once again. I wanted to see it, and there we stop. DP.
Yeah. So next we just have pictures of the certificates.
Oh, okay. Okay, that's fine. That's fine. So that's fine. Okay, so this is the type of certificate that we give, and this is for the last several years. I think we have been giving 8 years. We have been giving it. So that's so okay. So this will be the one that will be dispatched, and we will also try to find out the exact name and stuff like that before we send it. Okay, this is Saida, and yes. Okay, so that's fine. I think.
Yeah.
I think I took more than what you gave me the time.
That's okay. I think this is the recognition of lot of efforts that all of you have been.
And I don't know whether you noticed or not that I became emotional. I don't become otherwise. Okay, thank you. Thank you very much.
Yeah. Thank you, Arunan. And in the interest of time, I think we will continue with the rest of the program. And before that, I think Meena and Kiran wanted to just show something. Over to Kiran.
Yes, so I am uploading a PDF. Give me a moment.
We are just going to take a break and let Kiran upload that one slide.
Yeah. Now that there are 63 people, like we could also take a really big group photo if it is possible. Yes, once after.
So before going for the photograph, just relax and please follow this instruction. Just follow it for a couple of minutes.
Okay.
I mean, this is just an earnest request. We have been just sitting and listening. And after 1 or 2 minutes, DP, you can start the webcam.
Okay, sure.
Thanks, Kiran. So please stand up and do some stretching exercises.
Yeah, the webcam should show that they're actually stood up.
Yes. Good idea.
Then it's a real group photo, right?
So please stand up, feet together aligned with hips. Open up all the toes wider with gaps between each toe. Place the heels firmly on the ground. Lassie also wants to do that. Arms alongside your body, chin parallel, and look forward. Slowly lift your arms up, shoulders relaxed, some gap between arms and ears. Slightly lift your arms with shoulders up. Stretch, feel the stretch of arms upward. Now slowly lift your heels and try to stand on your toes. Look forward, chin parallel to the ground. Breathe normally, balance. Stand and feel stretch for 5 to 10 seconds. And then slowly place your heels on the ground, arms beside your body, relax, and can repeat for one more time. Thank you.
Okay, so what are we doing?
Anyway, my webcam is not uploading. No, it is just a break. You can go ahead and call for people with the screenshot.
This is live.
I think I'll have to kneel down for me to be visible.
This is not a punishment. Do some twists. Some side bends.
Yeah, this is a nice way to invite people to join. Turn on the cameras.
Great.
Yes, everyone, please turn on your cameras so that we can all take group photos as we stretch and relax. And I think you will naturally smile now when you are relaxed.
By the way, Kiran, can you get back the poster? Otherwise, the screenshot will come with this instruction. Get the first page.
We can minimize, no? The
Yes, I have
presentation.
minimized the presentation for myself. Yes, Rafiq, leaders. Yeah, GN is now going to stretch. You're muted. Yeah, by the way, all this is getting live streamed.
I did
FYI.
I did when the slide was before.
Okay.
So right now, if I lift, you stand up, this is what you see. and you can hear my lassi is also stretching. You can hear.
Yeah, we could invite her as well in the camera.
Lassi, come. Come. People are calling me. Come here. Come here. So no. She ran away.
Okay, more people. We just have 16 today. Yesterday we touched 18. More people, please turn on your cameras and keep them on. Yes, Arnan is also here. Arnan is giving neck twists.
Can't see myself.
Yeah, we can see you. We can see you. Ashish, Ravi, turn on your cameras if you are there. Jude, Farhan, all the awardees. Yeah, good to see so many of you virtually, though we couldn't meet.
Arman sir, you should stretch.
I should stretch.
He is stretched beyond imagination.
Okay, so should we get back? Yeah. Okay, so I think all of you have relaxed a little, and congratulations again for all the awardees. And we have 3 more sessions on zebrafish, nail regeneration, and then phyllanthus. So over to Saida.
Yeah, so It was quite fun, the exercise also. Not so. Yeah, coming back to the presentation, connecting Hydra with regeneration, earthworm with regeneration. We have yet another model system, zebrafish, which is working on regeneration and
Zebrafish, Denio,
an effect of toxic effect of embryo of zebrafish. Yeah, we can start the presentation.
radio. It is a small vertebrate. It contains around 80% of disease causing genes as humans. It has a transparent embryo. They have high fecundity rate. Nowadays, they have emerged widely as a model organism in various research studies, which makes them special.
Denio rareo, the particular species of zebrafish which we used as our model organism. Our short-term objective is to study and maintain the zebrafish. It was discovered by Francis Hamilton in the year 1822 and established as a model organism by George Stresinger in the year 1970. It is named as zebrafish as it has stripes like zebra. Why to consider zebrafish as a model organism? Because it produces hundreds of offsprings in weekly interval of time, shares 80% of disease causing genes with humans, also has a regeneration capacity, and the most important thing, it has a transparent embryo through which the embryonic development can be studied. How to identify male and the female fish? The male has a slender body as compared to the female. The male has a shorter caudal fin, whereas the female has a larger caudal fin. The adult female can be easily identified by the egg spot present on the body from where the female lays eggs. Classification of Denio rareo. The probes required for our research work were gravels, filter, anti-chloride and methylene blue, fishes, oxygen motor, fishing net, nutritive flakes as food, artificial plants, tank, thermometer, crucible with nylon net, bowl for breeding and pH paper. Our tank setup, which is 32 cm in length, 19 cm in breadth and 21 cm in height. Let's have a look on the life cycle of zebrafish. It takes 3 days post-fertilization for the egg of zebrafish to hatch and almost 90 days to become an adult zebrafish. Now let's have a look on the observations.
The length of the adult fish which was observed was 3.2 cm. It was observed that other parts of the body were not moving. The movement was shown only in the mouth region. Some fishes were swimming near the filter. Maybe they were not getting sufficient amount of oxygen and hence they isolated themselves from the entire culture. It was observed that the fish was unable to maintain a normal position. Sometimes the fish used to sink at the bottom or float at the surface or float upside down or float at the sides of the tank. The fish was not able to swim in a linear direction and started a barrel roll movement. When the ecosystem of the fish was changed, the fish used to show some aggressive behaviors. The change in the ecosystem means, due to the COVID-19 pandemic, the fishes were brought from the college lab to our residence. After bringing them to my house, some of the factors were changed, like exposure to the light, noises, new environment. Maybe these things affected the behavioral changes in those fishes.
Intermediate objective is breeding of zebrafish. For breeding, we took one male fish
and one female fish. They were kept separately for 7 days to increase the desire for mating. After 7 days, we kept both the fishes together in the same bowl for breeding. Here's a video of the breeding setup. For breeding setup, we took the ratio of 1:1, that is one male fish and one female fish. For the setup, we used a crucible and a nylon net so that the eggs would get collected into the crucible. But unfortunately, the male fish died and we didn't got the offspring. And our one male fish died. Long-term objective is to study the toxic effect in the embryo of zebrafish. Now, let's see how tail regeneration occurs in zebrafish.
This is the setup for washing the fish ball. Transferring the fish into different container with the help of fish net. Transferring the fish back into the fish ball after filling the ball with dechlorinated water. Finally, feed the fish.
Taking ice water into a cup, we'll select one male fish. We are taking this fish into the cold water, getting anesthetized in cold water. When we'll see that it is moving less, we'll put it into the petri dish and we'll add some normal water to it so that the fish doesn't die. Now before the fish, we can see the fish is still breathing. We'll add some more water so that the fish doesn't die. We can see the fish breathing. Now we'll cut the tail.
Many research is going on by considering zebrafish as a model organism, such as regeneration, embryonic studies, many different research in genetics, toxicity, and in the development of human drugs.
Goof-ups that we made during our work. First, we used the same water from the tank for the breeding purpose. Second, changed the ecosystem due to COVID-19, which led to some kind of stress in fishes, due to which they showed aggressive behavior. Third, we used large amount of gravels.
Our future plan is to breed the zebrafish and get the embryos and study the mechanism of regeneration.
Thank you so much for watching us.
It was quite fascinating, must say. So We can take up the questions. Over to you, Devas.
Just either you could take up a few questions from the chat.
Okay, so what is the role of the ice water and cold water before cutting the tails? Yes, that's it.
Yes, actually, the ice water or the cold water is used as anesthesia for the fishes before the amputation.
So how is actually this cold water anesthetizing the fin, as in, and then we are adding normal water to it. Can you elaborate a little on that?
In the beginning, we add cold water so that the body parts of that fish they get anesthetized, and then again we add the normal water so that the fish can again back to the normal conditions.
And one of the interesting question is, how do you identify male and female zebrafish, and why zebrafish? Like why?
Yeah,
For
okay. So
identification of male and female zebrafish, first of all, we look for identification of female fish. So the belly part of the female fish is enlarged, whereas compared to the males have slender body. And one more thing, we observe the caudal fin of the fishes. Female fish have larger caudal fin as compared to the male fishes. These are the distinguishing characters between male and female zebrafish.
okay, and why we choose zebrafish? The reason behind this is the embryos of the zebrafish, they are nearly transparent, which helps the researchers to examine the development which is happening in those embryos. And the eggs of the zebrafish are fertilized and developed outside the mother's body. So we can even it also helps in seeing the development which is happening in those fishes, and it has similar genes with humans. Around they have 70% of similar genes with humans, and out of that around 80% of the genes are disease causing genes.
So I'll take up some YouTube questions like, why do
Yeah,
we
sure.
choose zebrafish over earthworm and hydra? Because we have studied, like we got to know prior to this that there is regeneration in hydra and earthworm as well. So why should we go for zebrafish?
It is a small vertebrate, first of all, and second thing, it produces large amount of offspring, and the time interval for producing that offspring is that female can produce offspring at weekly intervals, and they produce around hundreds of eggs, even at first breeding. I mean, after weekly intervals, they produce the offsprings. So we can use that, and we can even get more number of embryos if we go for zebrafish, as they produce more number of the offsprings.
I will take questions from the chat. So there are a couple of questions which says the Anshu Kadam is asking like, how did COVID-19 affect your fishes?
Actually, COVID-19 didn't affect our fishes. We changed the ecosystem of the fishes. Like when the fishes were kept in our lab, the conditions were a bit different, like the exposure to light. Even in the morning, the fishes were exposed to light from morning 9 or 8:30 or 7 or 8:30, 7 or 8:30 to evening 6:30 or 7. And after 7, we used to leave the lab and the lights of the room were closed. But when I brought them to my home, since it's a house, I have to keep the lights on. Even exposure of lights can also lead to some kind of stress in those fishes because of the sleeping patterns of those fishes can change. And even in the college, we have, though we don't have much noises as compared to which we have in our home, like children are playing, even I have my brother and sisters, and the environment was changed. So this may affect the fish.
Can you
Why you
elaborate
think why
on?
Yeah, why you think the noise is going to affect the environment of the fishes or the development process of the fishes? And adding to that, taking one question from the chat as well, like you said that developmental studies could be done easily in zebrafish. Why don't you think other model organism like similar type of study can be done on nematodes as well? So why choose zebrafish and why over nematodes?
Actually, for
Uh,
developmental studies, we go for zebrafish because the eggs of the zebrafish are transparent. So, due to the transparency of the eggs, we can observe the developmental each and every developmental stages, and we can record as a observation. So, it is used in developmental biology.
Even
But
the
Akansha,
embryo,
similar type of thing we can study in the nematode as well, like C. elegans as well. So why to prefer zebrafish over C. elegans?
for zebrafish, like embryos are little large as compared to the nematode, and development can be seen with our naked eyes. And even the fecundity rate, like a zebrafish produce hundreds of offspring as compared to nematode, which might be less.
So, Kunal Kadam is asking in the chat box, why the zebrafish is looking reddish? What happened to the stripes? Are you sure that that is zebrafish?
yeah, that is a zebra
Can
fish.
you, can you tell me how to identify a zebrafish?
Oh yeah, they are being identified by the strips which are present on them. We use orange one zebra fish, and the strips were a silver in color, and they were all over their body. So this is one of the way in which you can identify the zebra fish because they have those strips on them.
So, any fish having strips would be zebrafish.
No, not any fish having strips would be zebra fish. It usually depends. Like uh, the the strips have they are spread all over the body, like from the gills to the last caudal fin, and not it is not the same in all the fishes.
One question from YouTube chat Kunal Kadam has put is like, how does light affect the development? And adding to that, there's a question from Anshu on the similar basis. It's like, why does light and noise affect the development of zebrafish? And the statement is there, but many people also keep their aquarium at home as well. So, any comment from the team?
Yeah, many people keep the aquarium at home, I believe, but if suddenly the environment is changed, this can lead to some type of stress. Like when I bought the fishes to my house, after 2 to 3 days, I've observed that some of the fishes were like fighting amongst themselves. I don't know the reason behind that, why they were showing that kind of behavior, but I thought that this may because ecosystem of the fish was changed. Maybe this can be the reason. Even when with the fishes in our college lab, we haven't seen such kind of changes or behaviors in them. So I said that.
But do you think that this is because of the space at which like the space total space of the tank that you are keeping in the college as compared to you have a less space of tank we are keeping in the home, or is it the
Um,
same space tank?
no, we have the same tank which college. We bought the same tank in our house. Nothing was changed. The only thing that changed was the exposure to the light. Usually, zebrafish prefer having the normal sleeping patterns, and if it is disrupted, then that can lead to some kind of changes.
It would be great if you can support this with some research papers or something, because just vaguely saying that light is having a role or is not playing a role.
Because in the chat Nagarjuna said when the fish were fighting in the college you were not there to observe.
Yeah, even that's possible. When they were fighting there in the college, we would haven't observed, but that's not the only reason. I've also seen some other kind of changes which we haven't observed in the college, like the fish was floating upside down, and fish was rolling around its back. Sometimes the fish used to settle near the filter. Only the fish used to swim only near the filter. I was wondering why is the fish is not getting engaged with the other fishes. So this I have observed these things just because of observing these things. I assume that because the ecosystem was changed, we lost our fishes, or the fishes were
So,
not
and
able to.
one more
Yeah.
question just to ask, like relate. I have read somewhere like if like is there a more number of males in the tank as compared to the females? So like for the courtship behavior, maybe the males are fighting among themselves. Do you think that there's a possibility of that?
No. Actually, in our case, it was opposite. There were more number of females and less number of males. Around when I bought the tank at my house, we had around four females and three males. Five females and three males. Sorry,
So
five females and
one
three males.
question again from the chat box I would like to take is, we saw in the video you were using
Yeah, so we
artificial
use artificial plants so that we
one
can.
at a
Yes.
time. Can we
so that it can create a natural environment for the fishes. Not We didn't use the natural plants because for that we have to make so many setups, and it was not easy for us, so we preferred the artificial plants so that it will create a natural environment for the fishes.
if you're using artificial plant, how is that creating natural environment for the
Yes,
fish?
exactly. And
Just
what basis you are selecting the artificial plant, or don't you think that if you create a like a natural condition, because zebrafish is not exposed to artificial plant, that will be more favorable for them to culture in the home lab?
Yeah, actually, we removed the plants after some days. We actually removed the plants after some days because maybe the plant was the color of the plant which we have added was fading, and even the water which was present inside the tank along with this artificial plants, the water became yellowish. And after when we removed the plants, yeah, the water was normal. So we removed that plants, artificial plants.
What is your research question? Because many people are asking what is a research question, and in your video they saw some red fishes which don't have strip. So is it the same Danio radio you're talking about, or is it some another species that you're talking about?
No, actually, those are the same fishes, but I think the color was a bit red. I don't know what was the reason behind that, but yeah, those were the zebra fishes.
is
And the research
Dano radio
question,
a species or I think I thought firstly like Dano radio is the scientific name for zebrafishes. Can you clarify?
yeah, it is. It's a scientific name for the zebra fish.
So under Dano radio there would be a lot of species.
Yes.
Okay, cool.
And our research question was to get the embryos of the zebra fish and then to study toxicology on that and developmental stages on those embryos.
Any study that has already been done on this? Any reference you have quoted? Because in YouTube chat, I think the speculations you made, Arunan sir is asking for any reference that you have.
Yeah, for that developmental stages, we have read some of the research papers, and yeah, I do have the reference for that.
Mm, can you
For the
quote some
development of,
paper? Can you quote some
yeah,
paper?
just a second, I'll do that.
Mm. Taking question from the chat, Arunan sir is asking how do we know that they are the same species and how do we classify zebrafish? Akansha, would you want to answer? Like Arunan sir is asking, how do we know that these are the same species and how do we classify?
The research paper which we have read was zebrafish development and introducing undergraduates to developmental biology in the embryos of zebrafish, and it was published on 2009 June 6, and it was published by Alison D. Costa and Lane T. Shepard.
Anshul, we want to answer the next question that I think in the chat it has been coming up. Like,
Can you
how do
repeat
you
that?
classify the? Yeah, how do you know that these are the same species, and how do we classify zebrafish?
We usually recognize zebra fishes with the strips which are present on them. Um, and
There are fishes that we see in the video. One is kind of resembling black and white, and one is sort of orangish. So
Actually, that was not black. Yeah. Just I'm adding to that. That was not black and golden. Actually, we were not getting a perfect picture for the identification of male and female. So we have taken that picture from Google, so that we can explain it in a better way. And the fish which we used was the orange species, like zebra denu of orange species.
Arjun sir is asking, is that a genetically modified zebrafish? And also to that, Kunal asked, puts a comment over here. As I search on the Google zebrafish, Danuario seem to be black and white. Are you sure the pink fishes are of same species? Why is the color difference?
Yes, there are possibilities that they can be the mutated ones. They are not the wild species. That is possible.
Okay, I have a question. When you talk about you want to study the developmental of zebrafish, like
Yes.
what are your expectation out of the study? What do you expect after doing the study?
Yes, just a second. After studying the embryological or the various stages which are various stages in the development of embryos, we can even correlate with them to the humans. The reason behind this is that, as I said, they have similar genes, and we can even study the concentration. We can even study the use of chemicals on these zebra fishes. It was our long-term objective, but unfortunately we didn't get the embryos, so we couldn't do all this.
Which
yeah, yes,
yeah, which chemicals you're trying to study, like the effect, because in, like, I will connect to my curriculum, because in our curriculum just now we've did a project, like there are like practical on effect. Is it similar to that type of chemical where you try to study different developmental stage, how it is going to affect or not?
Actually, we were trying to get it from kanamycin, as Saida was doing for the cardamine, and yes, for the cardamine. So we are planning to do it with kanamycin.
Why you think kanamycin is going to affect zebrafish, considering it is a eukaryotic plant and kanamycin is a eukaryotic animal, and kanamycin affects bacteria, which is a prokaryotic thing?
Can you repeat again, please?
Why you think that kanamycin is going to affect the developmental process of zebrafish, considering kanamycin is the antibiotic which generally affects bacteria, which is a prokaryotic, and zebrafish is a eukaryotic?
Akanksha, would you like to add anything to this? Akanksha?
I have a cross question to Bivas. Bivas, why do you think kanamycin would only affect prokaryotic cells and not eukaryotic cells?
Ma'am, considering if you see the how kanamycin works, it works on the bacterial ribosomes as compared to like bacterial ribosomes in the case of bacteria, and considering in Danuario we have eukaryotic organisms, or of course the ribosomal composition changes over here. So how can we be very sure before choosing a particular chemical that it's going to be as effective just to affect the entire development process of the zebrafish?
So for that reason, we
Kanamycin,
can use, yeah, please carry on.
you can say, ma'am.
For that reason, we can use other chemicals also. Because kanamycin was
Yes,
being
yes,
used, that's why you've
yes,
taken, or any some specific reason you've got?
yes. That's why my question is
Kanamycin,
like, why you're using kanamycin?
it affects the ribosome of the bacteria and the cell organelle of the animal cell, that is the mitochondria. It have bacterial ribosome. So I thought of using kanamycin, and actually we are also in future we are seeing that I have to study like what other chemicals can also be used.
Okay, what do you mean by bacterial ribosomes in mitochondria?
Omkar, please join the discussion. We have Omkar, one of the collaborators of Zebrafish. Please join the discussion.
Yeah, sure.
Yes, Hina ma'am, can you repeat the question?
Okay, I'll just, that means that 70S ribosome is present, just like bacteria in mitochondria and chloroplasts. You mean to say that?
So, as like we are studying the effects of kanamycin not only in zebrafish like Achal added, but also in cardamine and in rotifers we were planning, was because kanamycin, like Achal added, kanamycin could affect the mitochondria of animal cell because it has bacterial ribosome. It has bacterial origin. So yeah.
But my question is like, why we are trying to, so see, if we talk about doing it in rotifer, or doing it in the case of calamine, we are trying to figure out that whether the genetic, like we are using kanamycin to screen the thing, but in
Why
zebrafish,
kanamycin? Yeah.
yeah, but in the zebrafish we are trying to see the development. So why we specifically like banging on using of kanamycin? Like here we are not screening the thing with here we are not checking for any genetic engineering or stuff.
So that means you may hypothesize for some changes which you were expecting, the effect of chemicals on embryology or cell differentiation.
Why with different chemicals? Why just kanamycin? Like Vivas was adding about his experiments in college in its curriculum. We can try using that. So maybe Vivas can help.
No, I was just telling that why we use kanamycin in rotifer and kanamycin in calamine is just because we are doing a engineering and we are screening it. And but in the case of when we are doing a zebrafish study, then why should we use kanamycin? Like why not find out some reference which talks about, okay, these are the chemicals or these are the thing which are needed for the development of zebrafish, and if we can put some or the other, or if we can decrease or increase the concentration of some other chemicals out there, just like I am not very sure, but just like retinoic acid is one of the
Something,
things which,
yeah.
Why
we
this
can use that.
kanamycin?
Like something.
Why start thinking about different chemicals, but because of restriction of time, we have to stop zebrafish presentation as of now. We can take up the questions in WhatsApp chats for sure. This discussion shouldn't die, and this will of course help the zebrafish group to work upon their long-term objective. So as of now, we'll move forward with a nail regeneration. So earlier there was a question that can there be regeneration in humans? So seems like this group is coming up with nail regeneration. So yeah, let's have that presentation.
Greetings. My name is Apurva Rathod and I'm a student of Human Sciences. I study at VPN's B.N. Bangorkar College of Science, Thane. My study is going to be focusing on the rate of nail growth. I recall reading that the nails of our dominant hand, that is the hand that we use more, grow faster than the nails of our non-dominant hand, the hand that we use less. Now, my study is going to compare these two, the growth of the nails of these two hands, and I'm also expecting to find the difference in the nail growth between my little finger and my other fingers, because we use our little finger the least. The method that I'm going to be using will be, I'm going to get an imprint of my nail using paint. I'll demonstrate here. Like so. Then I'm going to imprint the same on a piece of paper, preferably graph paper. Like this. Then I'm going to compare the top of this with the imprint that I get after 1 week, after 2 weeks. That is how I'm going to compare the rate of nail growth. Thank you.
Hi everyone, I am Kunanika Ghosh from St. Xavier's College, Mumbai. I am a first year BSc student and my topic of research is nail growth, nail regeneration and the genetic aspects to it. As many of you might have noticed, older people have brittle and yellow nails which are prone to breaking. The reason for this is the increased levels of carbon in older people. This is suggested by the studies. My topic of research is one of these many factors which affect nail growth and nail health, that is the hormone estrogen. Women around the age of menopause, that is around 45 to 50, lose estrogen at a drastic level. Of course, not intentionally, but their body loses estrogen around the age of menopause. This brings me to my research hypothesis, which is women around the age of menopause will have lower nail growth as compared to men of the same age and younger women. Estrogens are very important for retaining water balance in the body, and water balance is very important for good nail health and nail growth, which is why estrogen is directly proportional to the nail health and thus nail growth rate. The methodology that I'm going to be following is monitoring 4 subjects. 2 of them will be around the age of 20 to 25, which is the age when women have very high level of estrogen. That is around the age of puberty, around like 15 to 25. And other 2 subjects will be around 45 to 47, and one of them will be a female and one of them will be a male. I will be observing these 2 males and 2 females every week for their growth rate of the nails. I will be recording this accurately with the help of a graph paper and indelible ink. I do hope that my research will shed light upon the direct relationship between estrogen and nail growth rate. I do hope our findings will help all of us know something more about nail health and nail growth. Thank you.
Was the nail regeneration experiment. For this experiment, the materials needed are mehendi and a graph paper. For the experiment, I had recorded the growth of nail of 3 people, 2 females of 47 years and 15 years and a male of 54 years. The female of 15 years showed uniform growth till the 3rd week, and from the 4th week, non-uniform growth was recorded. The female of 47 years also had uniform growth till the 3rd week, and from the 4th week non-uniform growth was recorded. But compared to the first female, the growth of nail of the second female is slower. The male of 54 years had recorded uniform growth throughout, but the growth of nail was slower than the females.
Second experiment was on nail regeneration. This aim was towards evaluate the nail regeneration in different age groups. The first was apply the mehendis in any one fingernail and observe it carefully and mark it in a graph paper. And then from these activities, it is also concluded that nail regeneration in different people is very different.
Of human nail regeneration. To evaluate nail regeneration in individuals of different age groups, firstly apply mehendi on fingernail completely, then observe it weekly and mark in a graph paper. Data was collected for 10 weeks from August. It had observed that nail regeneration is dependent on age. As the age increases, the rate of nail growth decreases. Thank you.
It was really nice video, and we have several questions coming up in the chat box. So one of the major, or we can say basic question would be, what is the difference between nail growth and nail regeneration? This is a question by Nagarjuna.
Okay, so I'd go ahead and answer that. Nail growth is basically the growth of the nail without it being amputated or being cut. Like it'll just grow uniformly, like not uniformly, but it'll keep growing even if you don't cut it, your nail. Whereas regeneration is even if you cut your nail, even if it gets broken or something, it will regrow. So that is the difference between regeneration and growth.
So how are you studying regeneration? Because there was a lot of methods seen, and are you studying nail growth or are you studying nail regeneration?
I think in this case, it's a misnomer in a way that we are saying that we are studying nail regeneration. Basically, nail regeneration and growth are the same thing. Like ultimately, you do cut your nails, right? So regeneration and growth here, I think we are comparing it on a similar basis.
So there were various ways by which you were actually looking for nail growth, if I may say. Um, so yes.
Yeah,
How are you?
Saida, for that we have actually created, like, as election time is around, and we are getting data from many people around. Like also a lot of Kochi schools has come up with some brilliant data, and also Sucheta ma'am from Goa is collaborating, and many people are giving us the data. So can you move the slide forward? We have some form similar type of we created a form similar type of what we created for mango phenology. Next. Yeah, so we created this form to study, uh, to collect the data from various places. So as we are considering the age as a factor, so we have put age as a factor. We are also considering gender as a factor, so we are putting gender as a factor. We are giving them any referable objects. We have given them like uh, a scale. You can take a 1 rupee coin, and also the date of the picture and the place where you're taking the picture from. And can you go to the previous slide, Kiran ma'am? Yeah. So here we mentioned that reference material can be anything: a scale, a graph paper, or a coin, or anything that you can study the picture from. And what do you need? You should have a watermark, or as like beautifully explained by the Kochi group, they have used Mehendi to study as well. So we can use a Mehendi mark as well. And also we are keeping a track of the data after every 7 days. So we can see that how much the nail has grown in the
Don't
7 days.
you think for Mendi, just one question, don't you think the Mendi will fade out after some time?
Yeah, that's
Yeah,
why
okay. So for
that's why
that,
I
we
haven't.
are preferring like indelible ink, and even if it fades, like the bottom mark will be in place. So the measurement that we are taking from the like bottommost place, it will be the same, even if the top of the mehendi fades.
Yes, and to also like many people may not apply Mehendi, and considering it is a citizen science project, so we will majorly target to the watermark after the voting, and we can circulate this form, and it is like And can you go to the previous slides? And yes, so we have shown that there are different methods to collect the data. Like we can collect data, you can put a coin, 10 rupee coin, and check it and place your nail right there, graph paper maybe. And this is one of the beautiful picture that is taken by that is given by Sucheta ma'am. She shows that how using graph paper we can also take the thing.
So there are various methods, like you can use any of them that seem suitable because we're targeting a large sample size.
Yeah, but just a question that how will y'all measure the size of the nail with the coin? Yeah.
So we are like using the precision up to a millimeter, I think. So a graph paper or a scale, I think, should be good enough for measuring the
And
size
also
of a nail.
coin will be good. Why I said, because these are the reference thing that is available in your home as well. So what you can do is like you can take measure the coin in your home and take a, like I say the coin 10 rupee coin has like 3 centimeter of diameter. I can check it for what diameter it is appearing in the picture. Similarly, we can compare that diameter with this thing and we can figure out like in the real life how much time the nail has grown. So on the basis of any reference
You might you can conclude it like you can make you can get approximate value, but I think with using a coin you'll not get exact value. You might not get. So just a suggestion that
why
no, I think we will get the accurate value.
Yes, exactly. Because why we are saying, because 10 rupee coin is the same size that you're getting in your home as well, and the person will be using it. So if I take the
Yeah, so
measurement
it's
of
like
the
a
10 rupee
standard.
coin, yeah, so if I so okay, I will explain you. If I take the measurement of the 10 rupee coin and came out to be 3 centimeter, okay, and when I'm taking the length in the picture, it is coming out around 3.5 centimeter because the person has zoomed the picture and taken the thing. Similar type of thing we can do it for, like we can relate it, like so suppose
so taking
so
actual coin for reference and taking as this picture and seeing comparing the both and getting the value.
Yeah, exactly. So we can know the actual size of the nail if we compare the size of the coin in the picture and the actual coin.
So taking up some YouTube questions, how is nail growth or regeneration same or different from earthworm or zebrafish?
okay, so nail growth or regeneration, if I talk about, we have other slides. Can you move a slide ahead? Some slides ahead. We have a paper. We have quoted one study.
Yeah, we have a paper about nail growth.
Yeah, just yeah.
No,
So
not this
yeah,
one, I think.
yeah, last this slide. We can talk about this slide as well. So we
Yeah,
in this paper, which talks about like why the nail growth or why the regeneration part is connected. So here you can see that in the nail matrix. So in the nail matrix, we have the nail stem cells which are present. So which is responsible for the growth of the nail as well as for the like not directly to the growth of the nail, but wind activation of the stem cell is responsible for the digit regeneration as well. So in zebrafish or in zebrafish or in earthworm also we are studying that how the regeneration process is getting initiated, or in case of hydra as well. So type of question like what signaling is responsible for the regeneration portion, what is the signaling that is giving the stem cell the cue to divide into a differentiate into more cells, can be answered to the nail regeneration concept as well.
Yeah, one thing I'd like to add is in case of earthworm and zebrafish, we were like studying those organisms. In case of nail regeneration, we are actually studying a human subject. Like we are studying the nail stem cells of a human subject usually, or a mammalian subject. So that's closer to human beings as much as we can get. Like the nail stem cells that we are studying are of actual the subjects are human beings.
So Kanika, what is the benefit of this study? What is the objective? If you are studying
The
nail
short-term benefit, the short-term objective is to study the effect of estrogen, like the hormone on nail growth, and of course consider the other factors too, like mineral intake, hydration, nutrition. There are many other factors which are responsible, so we have to consider them. So ultimately, there's a study that says that The nails themselves are the only parts of the body, they have keratin deposits, so they can actually like regenerate. So ultimately, this paper that is being shared on the screen says that with wind pathway signaling, we can actually regenerate the digits of a human being. So ultimately, it's ready for, and we can signal to a great extent. We can live to a
Okay, Kanika, you said that the nail consists of keratin. So even hair consists of keratin. Our hair, they also have keratin protein, if I'm not mistaken. And
Yes,
you
ma'am.
said effect of estrogen. Effect of estrogen. So even in males, we have had even in Kanpur we used to do this comparative data collection of an old person with a young person, and the old nails were brittle. So even in males, the symptoms of aging were showing in the nails. So even in
yes,
males,
yes.
the estrogen level affects.
Ma'am,
It should
to answer this
affect.
question,
Of course, it affects.
okay,
Yeah, Bivas, you
okay,
want to say something, or should I?
yeah. Yeah, I'm saying. So considering we are not only comparing the estrogen factors. So if you, as Kanika rightly mentioned, that we are also checking on various other factors. So it can be also possible that different medications or any supplements that the person may be taking may be affecting it, or the calcium content as well. Also, we have also found studies which says that hydration affects the development growth of the nail very much. So we have also in the form mentioned. So can you give a rough estimate of like how many liters of water you drink daily? So there's also a column. So Kiran, if you can kindly move to the form as well in the, I think, third slide of the
And the
form mentioned.
main thing is that why does estrogen affect nail growth? It is because it is known that estrogens retain water levels in the body. So indirectly, it's the hydration that is affecting. Estrogen is actually indirectly affecting nail growth in a way, nail
And also,
regeneration too.
yeah, also if you can see, we have put this as well, and we have took up this as well. Also, when we are discussing last time with Sucheta ma'am and Batul ma'am, they also that is it like completely male doesn't have any estrogen, or is it
No,
only
it's
the
not
female does?
that. The males do have estrogen, and that's another
Yeah,
important point. Males do produce estrogen.
and also it not
Yeah,
only
both the
depends on
hormones
like,
are present.
yeah, and
Yeah,
also it not only depends on the entire thing, like how hydration also. It also depends upon the what are the nutrients we are uptaking it. So we thought of like adding a column of like what you eat daily or what you prefer to eat daily, but considering that would be a very subjective
too
answer and it
subjective.
will
Yeah.
differ. Yeah, it will
So
differ. We
there
are
are
not
more
trying to add.
YouTube questions. How is sorry, how do we take care of variation in finger size?
Okay, variation in finger size. Ultimately, the nail growth, when we're taking up breadth might vary, but the length will ultimately be the same. Like if you have more nail bed, it will, of course, you will have a bigger nail. But ultimately, the measurement that you're taking of the nail will be of the length that it has grown, not the breadth. So even if the finger sizes vary, we can run a comparative study. I feel so.
And also what a study that I have found which talks about effect of finger length on hand dexterity, and that was supporting Apurva's hypothesis. We can also say paper also mentions that as we age, the nail tries to becomes flatter rather than like the proper convex shape we have. So maybe that also we can consider as well, and we have to think more about that. Yes, but like these are the studies we have found.
But
And
that is one of the
Bivas,
factors we have to consider too.
in the presentation, somebody was telling that the right hand has a faster growth rate. I think some term was being mentioned,
Yeah,
active
yeah,
or
yeah. It was about the
so,
dominant
right.
hand and the recessive hand.
And the
Yeah. So
recessive
how
hand.
do you say dominant hand means the right hand? Of course, the hand which we are using more.
Yeah, of course, that's a misnomer. It's not necessary that the dominant hand will be the right hand. Actually, it's not necessary that the hand we use most is the dominant hand, in fact.
So what is the criteria of your, telling that this is a dominant hand and the other is a recessive hand?
Actually, I think it's more about which fingers you use the most. Like, which hand do you use for most of your activities? I think that's
Yeah,
what Apurva
that's a
meant.
very subjective question, I think, Hina ma'am. Like because many people are, I have seen many people who can write with both hands. So considering we can't
Yeah,
say any of the
exactly.
hands are dominant hand, and
Yes.
why I think Apurva quoted this thing is like she wanted to check because she has read in some article, which we found out later that some studies has also been done. So as now I was talking about a paper which talks about uh, effect of fingernail on hand dexterity, which talks about that yes, there are some studies which found that there's a more growth nail. So if a person is doing maximum amount of task with one hand and let's say right hand and as compared to left hand, so they have given like finger wise analysis how the growth rate has changed. So which is very disruptive type of data because they have shown that the middle finger or index finger nail growth is more as compared to the other finger growth, which we haven't figured out why. And also there are some study which says that this type of studies doesn't occur. So there are many disruptive and conflicting studies that came out between like what type of growth we observe in dominant as well as the recessive hand.
And quoting another example, I'd like to take the example of Sachin Tendulkar, who was like a right-handed batsman, but actually he did all of his work with the left hand. So you cannot really say which one is the dominant hand and which one is the recessive hand in this case.
So that's what I'm saying. What is your criteria of deciding that this is a dominant hand? Is it
Actually, it was
genetic or is it
Apurva's
phenotypic?
it was Apurva's
Yeah, is it
hypothesis,
genetic?
and she is not
Like,
present here right now. So if she was here, we could have been able to answer the question better.
yeah, actually, Kanika, when we use the word dominant and recessive, it is basically used in a subject in heredity. But
Yes,
when
yes,
we
I
are
totally
using
agree,
it
ma'am.
here, yeah. So that's what I wanted to ask. Are you talking of phenotypic or you're talking of the genotypic? Like, is it
No,
something to
I
do
don't
with the
think it's
genes?
anything about the genotypic ratio, like because ultimately the
Yeah.
genotype will be the same. Dominance is
And
Okay,
basically
okay.
what we
It
use
may be.
more. That's it.
Taking more questions from the chat box. There's a suggestion from YouTube from one of the YouTube viewers. How about nail polish instead of using mehndi? So you
Okay,
Yeah, we
so
thought of that too. Even that's good.
yeah. Yeah, I will confirm. I will answer this because we talked about using nail polish as well as we can use permanent marker as well. But considering we need a lot of data to conclude something or the other study, we will focus. And as it is a citizen science based study, we will focus on the watermark because people. So if you tell me to put a nail mark and like and take a picture, maybe I would take, but somebody who is not like somebody will be willing to put a mark on his finger and take a photo after every 7 days. So what we can do is like all this process are possible also in the case of mehendi as well. But considering it a citizen science project, we will try to target this form and circulate this form especially during the voting season so that we can get maximum amount of data without convincing people that like oh put a mark on the nail and like collect the data.
Okay, so one of
Yeah,
the
exactly.
question is, how does estrogen affect the growth of nail in male and female of different age groups? And this is by Ritik.
Okay, that's a very detailed thing. I'd like to answer that. So, estrogen is basically very high in the females at the age of puberty. And as women approach menopause, the levels of estrogen drastically get lowered. So, that is why we expect that menopausal women will have lowered nail growth rates as compared to women around the age of puberty. As for men, men will have a constant amount of estrogen. Of course, that will get lower with age, but the only source of estrogen for men is the adrenal glands. So they don't have to we don't have to consider the menopausal factor here. So when we are running a comparative study, the age and the gender, both of these factors matter. Because of age, the carbon deposits get increased in the nails. So that is one of the reasons why nail health and nail growth like deteriorates. So we have to consider too many factors when we talk of age and gender and nail health.
And also like okay, I will cite one of the study because Arvind sir is asking for the references. So there is a study from the published in clinical intervening of aging, which talks about the effect of estrogen both in male and female. The study says the entire same thing which what Kanika said is a study on 2013 published on 2013. So I will share the link as well. And also we would like to like if there are any questions, and otherwise we would like to go through the form in detail, so that as Arundan sir said that there is a voting in Bihar, so we can probably circulate the form and
Yeah,
get
we
some
need
data
a
out.
really large sample size. So as many as people we get, it's better.
Yeah, so
I think
next
we
slide.
can go ahead with explaining that, and after that,
Okay, Bivas, please do that.
take one or more questions.
Yeah,
And
so
go ahead
yeah,
with the presentation. Yes.
yeah, so nail lesion study. So this is the form setup that what you should have is only a watermark, and you have to be like, we will be pestering you to send your data after every 7 days. Your data collected will not be disclosed with anyone on any grounds. It is only based on the pure study that we are doing it. Any reference material will work, whether it's a scale, whether it's a graph paper or coin or anything that we can get in our daily life. You just have to click a picture with a mobile camera and send it to us. Also, can you go to the next slide? What we want is your name, contact number, preferably WhatsApp, because we need to follow up with your data after every 7 days. Age, because we are planning to do something on the basis of age. We are planning to see the growth change in the age. Gender, because that's one of the most important thing, because we are considering the growth of the fingernail in the case of in the context of estrogen. Then date of the picture taken, because we have to keep a track. This is one of the reference picture we have put. So just by doing this, you can click any of this picture, any reference object, location from which you are sending, because it will be help us to map the data from which we are getting it. Also, next slide. Okay, so rough idea of like how much water you drink daily, because why we are using this, maybe because as we have found paper which says about the how hydration is going to affect the growth nail. So we have options from 1 to 5 liter and more than 5 liter, and also any supplements or any medication we are taking. And also one more interesting thing about what we got out of the entire discussion we made last day was Bartleman was suggesting if we can also contact any gynecological clinic, because people may be not open to disclose their menopausal age, like at which age they are getting the menopause. So if we can get some accurate data from the gynecological clinics, we can bang on our thing, like, okay, after this particular thing there's a drop in the estrogen level, so we are getting a proper we will get a result more accurately. So these are the things we are. And also, like, how are you sure you won't misuse my data?
Okay, and
That's a
another
very
reason that we are asking for the supplements and medication is because, as we mentioned about the calcium, like calcium will affect your nail growth too. And of course, if you're taking any hormone supplements like estrogen, so that will have an effect. So we are just like taking this data so that we can accurately know, and how you can know that we won't misuse your data, because ultimately we have no use of misusing your data. Like whoever asked that question, we are just doing this because we are not studying an organic any other model organism like earthworm, nematodes, or zebrafish. This is a human subject, and we cannot culture them at home, obviously. So we are not going to misuse your data. We just need to study that.
There was a
But
question by
that's a good question. Like, how are you sure you're not going to misuse your data?
So
We are just asking for data because we cannot culture human beings at home.
Okay, one last thing before ending this is that there was one suggestion by Kiran. I guess voting date isn't that important. Voting date, putting a date of voting date when they
Yeah,
do.
that's important, I think,
Yes,
because we
because
need to know
that's
the
what.
first date when the ink was applied.
And not only the first date, if we can, like, if a person gets the voting date maybe and he's on today and he sends a picture by tomorrow, so what we want is like we want the date. So there's an option in the form which says about like date, and every 7 days we are collecting the data. So date is very important. Yeah, voting date will be important.
Like at least the date when you're applying your nail paint or your indelible ink or whatever date, like nail polish and all, we are not focusing on right now. So the date is important. Of course.
And also what we can do is, like, Kanika, I think that is very good suggestion from Kiran. We can introduce one other question which talks about voting date, because if someone sends you the data after 7 days, and if we don't have the track of the voting date, then we won't
Yeah,
find
that's
out
true.
like
We
how
should
much.
introduce that question.
That's a good suggestion. We will incorporate that. Thank you, Kiran, for that.
And we will be circulating this form and we won't misuse your data in any way. So please give us the data. It will really help us in our research because we're targeting a large sample size.
DP is saying we are not trusting you. We won't.
Hello.
Yeah, Bevas, just remove that point then that we won't misuse your data.
Hello. Uh,
Yeah,
yes,
Prashant, you're audible.
so I am saying that if we do not get the voting ink, we can also use another method which we are discussing in your cube chat.
Yes, if you volunteer to do something like that, then we have no problem at all. We want as much as data as we can get. So if you are willing to apply some other ink mark and give us data, then please, thank you for that.
Because the same question came up while I and Kaninika was discussing. So for the for setting the referral picture, I asked Kaninika to just mark take a mark of the permanent ink and send a photo. So she immediately did because we are doing the study. But considering if I ask you to put that, will you be doing? So maybe most of the people will not be doing. So
Means I am
that's
saying
why we
that
are.
But if somebody has a
like
nail paint and she wants to study it like that, we are totally fine. If you're volunteering and you willingly doing that, then thank you for that.
means I am saying that some means or some means not like the nail painting or something on our nail. So I think I will send the data by my method by embossing my finger nail, and I will be accurately send you data.
So
And
that's
also
one
one method suggested by Suchita ma'am, she said that what you can do is you can take a you can use a tracing paper and trace your nail, the nail, and you
Yes,
can
yes,
plot that
I
entire
also.
thing that Prashant did, and you can plot the entire thing on a graph paper and see how much it has growth. So that's also one of the good technique that we can use in the study. But
Yeah,
considering
like
many
we can
people,
use any technique, considering that the data should be accurate. That's the only thing that matters. We were saying indelible ink because
Yes,
that would be
yes.
something that everybody would have.
So it was indeed a great discussion, and we are all looking forward to firstly vote and then send the data to Bivas and group for
and also we have some people from Bihar joining us. So currently, I have sent the form in the chat. So if they can just circulate among themselves, and we can get some of the other data out of that, I think that will be very good
Yeah,
because we
please
have
save
some
this
people
form
from
and
Bihar.
share it with friends and family and give us data as much as possible because we don't have any other option. We cannot find data from any other place other than everybody. Like it was collaborative study after all.
and also adding to that, people from the cubist, as we don't need to we have, I you will all be willing to give the data. So if you can put a name or man in your hand and just give us the data, so it will be also a great help because we have so many cubists joining from different parts of the country,
taking
so that will
one
also
last
help us.
question from YouTube, and then we'll of course be switching on next presentation is, does drinking more water help in nail growth?
Yes, of course. I'll say yes, of course. The nail growth and nail health will be better. Actually, any keratin deposits, your hair, your skin, your nail, will be better if you're drinking more water. Hydration is one of the major factors, and estrogen is indirectly hydrating you. Like that's the thing. Yes, of course.
Okay, cool. So, taking a break from regeneration, we are coming to Philanthropist study now. So the Philanthropist group will be talking about sleep-wake cycle of Philanthropist plant and effect of different color of light on sleep-wake
Why
cycle of
Philanders?
Philanthropist plant. Yeah, let's start.
Why Philanders we taken as a model system? What is the important in this plan? These are found in all over India and it's also easily available at everywhere. Philanders plants are known for medicinal properties. Philanders plants are growing as a weed. Philanders are known as different local names.
In English, gooseberry, Hindi, boovi amla, Malayalam, കിഴാർനെല്ലി, കിഴുക്കാനെല്ലി, Tamil, കിഴാനെല്ലി, Kannada, നിലാനെല്ലി. Philanthus plants have very small life cycle and they are completed their life cycle 60 days. Other than medicinal properties, what else? Leaves are opening at morning and closing at evening. Is it touch me not plant? This is not touch me not plant because while one touch it's not closing their leaf.
Identification of the philanthus is very interesting at the same time very challenging too. As per the Bentham-Hooker system of classification, which is widely followed in India, philanthus belong to the family of Euphorbiaceae. So let's look at the taxonomic tree. Philanthus belong to kingdom Plantae, division Spermatophyta, subdivision Angiospermae, class Dicotyledonnae, subclass Monochlamydiae, series Unisexuals, family Euphorbiaceae, and genus Philanthus. According to recent phylogenetic system of classification, Philanthus belong to the family of Phyllanthaceae. Most of the people are familiar with the gooseberry. Actually, this gooseberry is belong to the same genus Philanthus, and its scientific name is Philanthus emblica. The other species in Philanthus genus are Philanthus amarus, Philanthus urinaria, Philanthus dabilis, Philanthus acidus, etc. Philanthus is a Greek word which means phylon as leaf and anthus as flower. We can easily identify the Philanthus plant which is having flowers and fruits under the leaves. Phylanthoid branching is one of the most important character that the branchlets resemble pinnate leaves with limited growth. And also the Philanthus plants having no latex. Leaves are simple, which is having flower buds on each leaf axils. Also, the leaves are alternately arranged and the leaves are having smooth margin. The cataphyls are the leaves on the main axis which are reduced to scale leaves. Stipules are actually scale-like structures which are used to protect the leaves when they were young. What is Monochlamydiae or perianth condition? Normally, when we take a hibiscus flower, it has mainly 4 whorls: calyx, corolla, androecium and gynoecium. Calyx is the green colored part and corolla is the red colored part, which are the petals, and they together seen as 2 separate whorls. But in case of Philanthus, calyx and corolla are not distinguishable from each other and they are together constitute the perianth condition. Also, each of its members are called tepals. The inflorescence of Philanthus is called cymules, which means cluster of male and female flowers are seen on the axils of each leaves. Flowers are unisexual, which means the plant having male and female flowers separately. Male flowers having stamens 3 or 6 and female flowers having carpels 3 which are united and also having. The fruits are actually called capsule. It is a dry dehiscent fruit. After drying the fruit, it gets burst out. The fruits are developed from tricarpellary syncarpous superior ovary. A fruit having 3 compartments and in each compartment contain 2 seeds. Seeds are trigonous in shape. Longitudinal ribs are seen on the back side of the seeds and seed coat is very hard.
We can go through identification part. We identified 2 species of Philanthus, Philanthus urinaria and Philanthus amarus. They are commonly distributed. These are identified on the basis of morphological characters. Philanthus urinaria having angular and pentagonal stem, Philanthus amarus having round stem. Leaves of Philanthus urinaria respond to touch and in amarus it will not respond to touch. Urinaria having asymmetrical leaf base and amarus having symmetrical leaf base. Leaf apex of urinaria is pointed and also having hairs on the margin of leaves, and that of amarus is rounded and no hairs on the leaf margin. We can see many flowers under the leaves. So we can count the number of tepals. The number of tepals in urinaria is 6 and that of Amarys is 5. Urinaria having 3 stamens that are free at the tip. In Amarys also 3 stamens but they are united at the tip. Fruit surface of urinaria is rough and in Amarys it is smooth. These are the characters used in the identification of these two species.
Hey, Philandus niruri.
Don't call me that. I'm Philanthus amaris.
There are many misconceptions regarding the identification of Philandus species. Most of the people call Philandus amarus as Philandus niruri, but the actual fact is that these two are two different species. For knowing the actual fact, we have to go through the taxonomy history. Philandus niruri in the book of Flora of British India written by Hooker in 1887 is described actually as a mixture of three closely related but distinct species, namely Philandus amarus, Philandus debilis and Philandus fraternus. These three species are together called niruri complex. Does light plays any role in sleep-wake cycle? We can do an experiment by placing Philandus plants under continuous light. We can make a hypothesis that sleep-wake cycle of Philandus urinaria disturbed under continuous light. The reasoning is that normally Philandus urinaria wake up during the time of sunrise and sleep up during the time of sunset. Let's know more about the experimental setup. We have used LED bulb for providing the continuous light. And also we have kept 5 Philandus plants in a room by providing 24 hours of continuous light. And we have placed these plants for 5 days. We have also observed the wake up and sleep up pattern of Philandus plants by comparing with the plants in normal habitat. Based on the observations of 5 days, we have plotted the graph by comparing the Philandus plants in continuous light and the plants which are kept in normal habitat. Surprisingly, we have got an unpredictable result that even though the Philandus plants which kept in continuous light, they continued to open and close their leaves. At the same time, by comparing the normal sleep-wake time of Philandus plants, the plants which are kept in continuous light show a delay in their wake-up and sleep-up time. The major conclusion that we reached from our experiment is that light does play some role in synchronizing the sleep-up and wake-up time of Philandus plants. At the same time, the Philandus plants in continuous light is continue to open and close their leaves, which means some other internal factors are controlling the circadian clock or sleep-wake cycle of these Philandus plants. The goof-ups of our experiment. The first one is that we does not keep a control inside the room by providing normal light and darkness. And also we forgot to observe the plants which are kept in continuous light after keeping them out after 5 days. And also we forgot to measure the intensity and voltage of LED bulb that we used. And the other thing is that large Philandus plants are used instead of using small ones. Our major fault is that we have used less number of samples.
My expectations or hypothesis for this experiment was that the Philanthus plants which are kept under different color of light will follow the spectrum of visible light that is absorbed in order to close their leaves or in order to sleep. So for example, the plant which is kept under red light will close its leaves earlier as compared to yellow, green and blue, because the red color light has a much longer wavelength as compared to the other three. Second, the plant which is kept under yellow light will close its leaves. Third, the plant which is kept under green light will close its leaves. And lastly, the plant which is kept under blue light will close its leaves, because the blue color light has a much shorter wavelength as compared to red, yellow and green.
I am observing my Philanthus plant, and it has been observed that my Philanthus sleeps at 6:30 p.m. in the evening. What about you, Lakshmi?
My plant sleep at 6:30. So why this happens? Both our Philandus plants are same. Then why they are sleeping at 4?
Maybe because we are at different places.
Then we have different in sunset time. So, Kiran and Lakshmi, when you are considering the sleeping time?
I consider when my plant started to close its leaves.
But I consider when the plant completely closes its leaves.
Both have different considerations, is it?
Yes.
So guys, can we do titration here as we do it in chemistry?
Oh, how?
As we do in chemistry, we titrate till we reach the end point.
Let's find out the end point of wakefulness and starting point of sleep. Only one leaf was taken for observations, and photo of the plant was taken at 1 hour intervals from 7 am to 5 pm, at 10-minute intervals from 6 am to 7 am and 5 pm to 6:30 pm. Angle was measured in between stem and midrib of a leaf. Angle was measured with the help of an online protractor. Photo of the plant was uploaded on this website. First pin was adjusted on stem and second pin was adjusted on midrib of a leaf. That is how degree of an angle was shown.
By measuring the angle at different intervals, we found out that between 9 am to 5 pm the angle is not constant. But this is just a 3-day data. We want to further continue this study so that we are able to conclude the time at which Phyllanthus actually sleeps.
Philanthus plant's leaves are opening at morning and closing at evening. Can we see the fruit flies also show same rhythm? Active at morning and sleeping at night? The 2017 Nobel Prize in Physiology or Medicine was jointly awarded to Jeffrey C. Hall, Michael Rauschbach and Michael V. Wong for their discovery of molecular mechanism controlling the circadian rhythm.
Indeed, it was great video, especially the part where we are kind of collaborating, showing collaboration between Fruitfly and Phyllanthus. That's great. We'll start taking up questions. So
before that, we have another video. We can play that. It's a short one.
the time lapse video was really nice, Vishnu. So we can continue with the presentation and start taking up questions from the chat box. So do we have any reference about that the Phyllanthus doesn't belong to Euphorbiaceae family anymore? Anyone from the group?
You are now. Could you please repeat the question?
Do we have any reference about? We can have, yeah. Do we have any reference about that Phyllanthus doesn't belong to Euphorbiaceae family anymore? Could you hear that? Hello?
You are now. Hello. According to the recent classification, Phyllanthus is not belong to Euphorbiaceae. It belongs to Phyllanthus family. there was a reference with the systematic studies in herbaceous Philanthus species. So and the region of Tiruchirappalli district in India. In this reference, it was clearly says that according to the recent classification, that is mainly APG system of classification, Philanthus is belong to the family Phyllanthaceae, and now
And
it is not.
also adding to that, there is another paper from American Journal of Botany which talks about phylogenetics of Triphylanthus, where they clearly mention that Phyllanthus doesn't belong to the Euphorbiaceae family from the recent classification point of view.
So, can we have the reason, like, why suddenly there's a change now? What did they find out that was not fitting into Euphorbiaceae family? Hello, am I audible? Hello.
Yes, the recent classification is mainly based on the genetic level. So according to that, so the family of Euphorbiaceae is actually split into different families. Such as Phyllanthus and Phycrodendraceae and other families are shifted. It's mainly based on some genetic level basis. Some of the base pairs which are common to some plants. So it fully based on instead of morphology. Now the classification is mainly giving importance to the phylogeny.
Lakshmi, can you tell me? So there's a YouTube comment. Is touch me not plant not at all related to Phyllanthus plant?
You are now. Actually, the Phyllanthus plant is not like a touch me not plant, because in case of touch me not plant, that is the mimosa plant, when we touch this plant, it immediately closes its leaves. But in case of in Phyllanthus, when we consider the Phyllanthus, there is only one species showing this character, but it's not just like mimosa plant. We have to repeatedly touch. When the raindrops fall onto the leaf, it gets close. It was Phyllanthus urinaria, but it is not exactly like mimosa plant or touch me not plant.
And
But it
also touch me not plants doesn't actually show the circadian clock.
If one person is speaking, others can mute their
Yeah.
mic so that there is no echo. Yeah. Yes,
Yeah,
Siva.
so touch me not plant doesn't show the circadian clock, whereas only one Phyllanthus urinaria, which shows a similar type of characteristics as that of mimosa family. Otherwise, the Phyllanthus are like as Lakshmi clearly mentioned in her studies as well. It shows a pattern of closing leaf and opening of the leaf. So that type of circadian rhythm is not shown in touch me not plants.
So why do we study Phyllanthus? Like, how are you choosing Phyllanthus? What is so unique about Phyllanthus plant? If I'm
So, talking about Phyllanthus plant, I think Lakshmi will add on to it more. So Phyllanthus, you can easily spot this plant in any, like, it is accessible, like as you can say, like any one of you can spot in any of the places. It's a type of weed. I would doesn't say because I'm not confirmed that whether it is a weed or not, but it is already available in many of the places. And also, considering about circadian rhythm. So what is circadian rhythm? As you know that few years back, Drosophila circadian rhythm got a Nobel Prize. So similar type of circadian studies we can do on Phyllanthus. So we can study Phyllanthus for that reason. If any Lakshmi would like to add more.
You are now. These plants actually showing it's actually a sweet, and the specialty is that very it having a very from the very first branch onwards it has developing fruits. So so having very short life cycle, and also the plant which is more interesting to study the sleek bake cycle is that the leaves of this plant which open during the daytime and also close its leaves in the evening time. So it shows this similar pattern is repeating in every day. Like we all know that there is a sleep wake cycle in all organisms. We also have a sleep wake cycle, and also when we can see more the daytime which was we can see the birds are chirping in the morning, so the butterflies are only seen in the morning, in the daytime, and they cannot be seen in the night. And likewise bats are more active during night time, and they are not active during the daytime. Like that these plants are active during morning time, and they close its leaves in the evening time. So they are showing a rhythm, and this rhythm is continuing every day.
So everyone is kind of surprised by the time lapse photographies, time lapse video. So in the YouTube chat, a lot of people are asking, how did you manage to do that time lapse photography or time lapse photo?
I think time lapse photography was taken by Sushant Tanti sir from Assam. He's one of our collaborators who's collaborating from Assam. I think I'm not very sure about how he took entirely, maybe putting his mobile in front of that, but considering he's not there, so I think he will be the best person to tell how he
Yeah,
has
we
taken
can
the photo.
definitely follow it up in the group. So there's another question regarding Philanthus. Like people are comparing Philanthus is also like snail, because do we plant wake up response to sunlight using the same molecular mechanism as animals?
Yes, we can actually, that's a very good question. I was looking at that from Ashutosh sir. It's a very good question. Yes, we can actually compare. Yeah, there will be, because if you see that, Lakshmi, do you have a slide for the titration thing that you did? So if we can go through that slide, we can.
Yes, Drishtant will show.
Okay. So if you see the titration data of circadian rhythm in the phalanthus, you can see that over the period of like 6 a.m. to 7 a.m. there's a like opening of the leaves. So basically, when the initial sunlight or like a sunrise to a particular thing and the sun rays reaches to the plant, they are opening. So maybe some molecular mechanism there are which is similar to that of the what we are answering in the case of Drosophila. So we can also take this study as to find out, is there any differences in the molecular mechanism of how the phyllanthus, the molecular mechanism in the circadian rhythm of the phyllanthus and in the case of the Drosophila.
So do we have any literature to support how does the plant respond to light or not? Do we have any research paper or any literature upon that? Was I audible? Hello?
Yeah, so there are many research paper which talks about, so like I've not gone through the paper entirely, but there are some papers, like one paper in the Plant Cell American Society of Plant Biology, which talk about the plant circadian rhythm. They have mentioned the molecular mechanism of how it does, but to be very honest, I haven't studied that, so I won't be able to explain in that much detail. Yes, there are papers which we are following and which we will be discussing while we talk about this.
So we kind of looked upon how the titration is made. So can you like elaborate upon it? Like why do we need titration? Though it was very clear in the video, but still like giving more importance to it. Yes.
So talking about plant circadian titration thing. So uh, I had some initial conversation with Kiran, and Kiran also, if you want to pitch in, you can come and join in. So what we said that it's like what we do in the chemistry lab, just testing like at the point. So we need to decide a point that where the exact change is taking place. So earlier when we are using the when we are getting the data from like different parts of the country, which talks about, okay, some says that it's 6:30 the plant is opening, some says that it's 6 o'clock the plant is opening, some says that it's like 5 o'clock the plant is opening. So we want to actually uh, find out the range at which point it is opening, or what is the range time, because there are different data from different places that we are getting on a daily basis. So then I think Lakshmi came up with this idea of using online protractor, and that is one of the good finding of Lakshmi. I would really say that, because that made Lakshmi's work more easy, because she could easily find out the degree or angle at which they're opening. So if you see the data what she has got from the titration thing, it's like shows you a range right from I think 5:30 or 6:30 the how until 6:30 how it is opening, and from angle like from 0 to angle 100, so how they're opening. So I think that's one of the greatest finding. And Lakshmi, if you want to add something to that.
Um,
So, I remember sharing a paper in interaction between light and circadian clock in regulation of CA2G expression in Arabidopsis. So, and this is a finding by Jeffrey C. Hall, Michael Young, the same scientist who worked on Fruitfly, got Nobel Prize on circadian rhythm of the Fruitfly. So it's great that we are kind of collaborating with the Fruitfly group. And so sleep wake cycle is not necessary. We can only study by Fruitfly. We can also study by Philanthus. And someone in the group was mentioning that how do they respond to the stimulus? We wonder if it is a spinal cord or not. So yeah, I think it was DP. So there are a lot many questions and lot many discussions coming up. We'll surely follow it up in WhatsApp group. So yeah, over to you, DP. Hello.
So Lakshmi also mentioned about that she is identifying a species of Phyllanthus. And in the movie also we saw that there are two different species that she is looking for. So any reference for that, Lakshmi?
You are now. Yes, actually, it having that is mainly a comparative study of this molecular and chemical characters of this plant, both by comparing these two plants, that is Philanthus amaris and Philanthus urinaria. So, can Arundan sir please come and talk about this presentation that we are having for the past 3 days about the national mean and conclude about it. Surely we are going to discuss it further in WhatsApp.
Yeah. Sorry, I was there on the YouTube and I was speaking muted. Yes, Arnan, please, if you want to say something as we close this session. Yeah.
Hello. Hello.
Yes, Arnan.
I thought we never close.
Yeah, close for the day.
Close for the night.
Here. Yeah, close for the night here.
So
we always continue conversation.
I wrote somewhere in some chat, I think I wrote that I think we always have got this tradition of continuing the chat at 12 o'clock, 1 o'clock, and round the clock. So which I think we need to, like Saida was telling, I think we need to continue in all instant messaging systems and all the messaging systems. So that's all. So it's only one of the days. Though I think we had a fantastic effort from all sources, and it was absolutely brilliant. The collaboration is absolutely brilliant. Thanks a lot, DP. I think please carry on. Yes.
Yes. Thanks, Arvind, for your words. And GN, do you want to add any comments or suggestions? I think all the students have been really wonderful, have put a really wonderful effort. Yes, please.
Yeah, can you hear me?
Yes, we can.
Well, I mean, there is nothing. So we have reached another landmark. I think, as you can see, in any pathway and a road, you have landmarks, but the road continues.
Yes.
So we are on the way. And so is Kiran around?
Yes, she's here.
And so, so Kiran, I think now the season 3 starts from tomorrow. And yeah, so and then we keep counting. Again, I think so. We have sort of concluding season 2 right now of the Chatshala. And in fact, maybe what we can actually do is that with every season, we can have a short concluding presentations of this kind.
Yeah.
And I want to encourage people to keep making videos the way how you have been making. And anytime, there is no need for a conference. You know, whenever you want to say something in a short time, video is a very nice medium to also express your creativity and also write a script once in a while. So what I would suggest is keep making videos, even if there is no conference, and upload them on chatShaala.in, and keep generating such resources so that you know, the undergraduate colleges as well as schools will start using these resources as their regular learning materials, so that people will start not reading science textbooks, but then they will start reading and watching and learning and participating in Chatshala as a model of doing science. So we have a long way to go. And of course, I don't want to name anybody, but I think all the Cubists, thank you so much for doing such a wonderful thing. And it's actually terrific. We didn't expect that even during the COVID, we will be actually doing uninterrupted chatshalas. But actually interesting, I mean, most interesting was that before COVID, our chatshalas and causeries were interrupted. Of course, online discussions were always happening, but we were not really doing synchronous meetings when COVID was not there. And what actually happened, of course, was that we invented a new model of causerie or a chatshala, and that happens almost every day. And I think that is thanks to COVID that you helped us to reorganize ourselves, and we are more organized now. We can even fight you much more organized way. Together we'll be able to face you as well. Even if you mutate, doesn't matter, we are also capable of mutation. And that is what we call ourselves learning. So we'll change, we learn by changing our strategies collaboratively, and that is how we will be fighting or struggling to learn STEM as well as rediscover new ways of tackling the problems of the world. So season 3 begins from tomorrow. Kiran, so take over, and back to you.
And I would like to invite all of
And,
you.
and Kiran has been the captain of this ship that is sailing forever. So Captain Kiran, take over.
Thank you so much.
Captain Kiran.
And I would like to invite all of you, like YouTube viewers and spectators here in Big Blue Button, to our season 3. And this is an open invitation to all of anyone can join us. We meet every evening, 5:30 to 9:30.
Thank you, Kiran. Yes, Arnan, you wanted to say something. Arnan, were you trying to say something?
No. Thanks.
Yeah.
Great.
Okay. So I think that's the beginning of our season 3 from tomorrow. Thanks for staying with us all through, and see you all immediately after this session through our various chat modalities. Thank you, everyone. Good night.
Can we take a group picture before ending?
Yes, we could take, if you all want to, but I think we can stop the stream now and we could continue these interactions.